I think we're going to get started. I want to say good morning and thank you for being here. We've been doing this for several years now, and every year the attendance goes up and the engagement goes up. So I want to thank you all for coming. We've tried to put together a program that is educational and informative and kind of spans the spectrum of kidney cancer interest, from localized kidney cancer, surgical concerns, advanced kidney cancer concerns, but also some of the ancillary concerns that we hear you guys all express about nutrition and lifestyle and how we can all be healthier individuals. So I want to thank you for being here. Dr. Shuck was nervous about filling an hour, so we made the excuse of everyone kind of filtering in late to give him less pressure for time. But we're lucky in this event that for the second year, we've been able to bring in a visiting keynote speaker. And I have the pleasure of introducing Dr. Brian Shuck. So I've known Brian for over 15 years, which is a little crazy because I know you look at me and you're like, but you're like 19 years old. I know. But we've known each other since we were very young. Brian and I were residents together at UCLA, and then he went off to the NIH to do fellowship in urologic oncology. Brian was at Yale for many years and was recruited back to our training institution, UCLA, where he's the director of the kidney cancer program. He holds an endowed chair in kidney cancer research. And in our community, which is a small community, Brian is really known as the leader, the guy in the genetics of kidney cancer. He has written numerous articles that have been featured in incredibly high-impact journals like Nature Genetics, Clinical Cancer Research, the Journal of Clinical Oncology, about the genetics of kidney cancer, and it's something that he feels particularly passionate about. In the last year, Brian got a notable accomplishment of being one of the co-leads of the kidney cancer studies within the South, well, it's not called this anymore, within SWOG, which is the big network that puts together large-scale clinical trials in kidney cancer care. And so in the next 10 years, we're going to see Brian's name as a leader in some landmark trials in kidney cancer. So we are very excited, very honored to have Brian give our keynote address on the genetics of kidney cancer. So thank you very much, Dr. Shuck. Thanks, boss. Thank you. Is that you, Brian Schultz?
Okay, thanks, John. Thanks for having me here at this great event. My first time in Seattle, actually, and I only visualize Seattle from my favorite movie, which I've watched like 30 times, War Games, with Matthew Broderick, and it looks a little bit different these days. So thanks, John. So I'm going to basically go over the genetics of kidney cancer, and I was told not to put any fancy kind of graphs or slides, and it's kind of hard to describe genetics without having some information. I'm going to kind of go over things in moderate detail, and the disclaimer is it's challenging, so there are some complex terms which I'll try to kind of make more understandable. And really also disclaimer is that half of what I think I know is probably incorrect, and I don't know which half. So we're going to go over really an overview of terms, really describing what genetics is, really focus on, the germline or the alterations that someone may be born with, and also somatic, the ones that are kind of acquired that are later during one's life. And they're kind of relevant to kind of different populations of kidney cancer patients. In terms of an overview, well, every cell in the body has basically a central area where all the genetic code is, the nucleus. And basically all the genetic information is wrapped around proteins on these things called chromosomes. Every cell has them. And these chromosomes basically have kind of genetic sequences, or basically the fine code which tells a cell what to do, which is the DNA. And on that, there's some genes. Every cell in the body has these chromosomes. Most patients, other than some complex genetic conditions, have 46. And men and women have different kind of sex chromosomes. But basically, we have normal numbers of these in the cells. And these can become abnormal in one's lifetime, or actually just in development. Again, all these complex chromosomes are wrapped around. There's a genetic code DNA, that makes up what's called a gene. So really, we have a cell. In this cell, there's the brain is the nucleus. In there, there's chromosomes, and there's genetic information. I'm going to basically explain how this genetic information can become incorrect and lead things, just like a computer program, you can have a change and you can have a virus on your computer and your computer won't start. And basically, we have this just alphabet telling the computer or telling the cell what to do. Sometimes this alphabet can become distorted and it can lead to bad things happening. So really, when we talk about genetics, a lot of it is basically this alphabet gets jumbled and we can't make sense of these words. Basically, when we talk about what do these genetic information mean, well, you have a word and it's told there's a meaning. Just like here, you have a DNA and a gene, and it is translated down to what's called a protein. It's basically the word is telling something to happen. And we have normal genes which code for things like hair color, for things like height. There's genes that control earwax. My wife, she's a different ancestry than me, and she doesn't have earwax, and she says she doesn't smell. And there's actually a gene in the Korean culture which actually there's patients have an alteration, and there's just less body odor and there's less earwax. I went to Korea, and there's actually no deodorant at the store, and there's actually a gene, if you look in a recent science paper, which may be altered in certain populations, and patients just have flaky earwax, and they have less body odor. My wife's blaming me because now my child is probably going to have bad body odor and wet earwax, and of course it's my fault. But this can also, these genes could go awry, and you can have something like cancer risk, okay, or basically risk of cancer. So genetic variation is normal. Everyone has, we have billions and billions of bases. There's about 28,000 genes. There's a little bit what we call polymorphisms or variations, which make someone different from someone else. So I might be not attractive. John Gore is very attractive. We have slight variations that actually are different in us, but that doesn't mean it's bad, okay? Natalie and Stephen might have a difference in their genes, and these genes might be seen frequently differing in the population, and we call that a polymorphism. Basically, these are giraffes, okay? We all recognize them as giraffes, but there's differences in one giraffe versus another giraffe. I would tell you, I went to the zoo, and my daughter recognized that they're all giraffes, okay? But these giraffes are all a little bit different. There's a little bit of slight variation. This is with her doll, Sophie, and they're all related to potential some variation in genes, which are not bad, but they're just making us who we are. So sometimes change is bad, okay? So changes that occur greater than 1% in the population are really what we call kind of just variants or polymorphisms. But, you look at Carrot Top, he looks very different. Sometimes, things go awry. Willy Wonka has been remade, and that, I would say, is not such a good representation, even though Johnny Depp is very good. So not every change is good, not every change is bad. But basically, when you have bad changes happen, instead of having that normal genetic code, that alphabet, that word, lead to a good meaning, it can lead to either no meaning or maybe the word doesn't actually get spoken. This basically of how changes or mutations happen and how they can change context. So I'm in LA, everyone wants to be an actor and actress. So let's say John is going for an audition and they're trying to say, hey, next up, John Gore, come in and speak your part. Well, here's just some examples that, a word may change. So if I change John Gore to John Gore with 2 R's, who's actually, if you Google him, is actually very famous, that is what we call a missense or an alteration, which is not really changing the meaning, and John will still walk in and hopefully get the part. But Sarah Suka, if we take out the couple letters from a word and say Dr. Sarah Pa, she may not turn around, she may not audition for that part. Or Dr. Dan Lin, you might add a lot of I's in there, and he may not recognize that or might jumble things. If John's coming in and reading on a screenplay, trying to become, get called, if you're reading a part and Jack is reading a part and he basically has his part of the script, let's say you get rid of that, the area saying, it's actually Dan's part. Okay, let me go back, sorry. Then Jack actually may continue reading into someone else's lines, okay? So that, you're gonna be upset, you may actually not get the part because you're reading someone else's lines. So there are a lot of these alterations, and not every alteration or mutation is bad. So again, there's changes that happen. It can be at the start of the word, it could be at the end of the word, and in a word, it could be at the start of the sentence, or it could be at the end of the sentence. So there are different changes, and all of them have kind of functional kind of consequences. The way the genetic code is understood is in patterns of 3. So a 3-letter word like the cat and the dog ran for fun, if I take one letter out and everything shifts over, then the whole sentence is going to be very hard to understand. Right over here, you get rid of the C and everything shifted over. So a whole gene, you may not understand that sentence because it's read incorrectly. And the role of the gene in cancer, in LA, pretty much everyone has a Ferrari but me. I moved about a year ago from New Haven where, we were a Honda, we were like a Chevy, different Toyotas, and everyone has a Ferrari and they're all sitting in their traffic. And, cancer is basically these words get changed and this car gets into an accident. And there's basically two types of changes that are really bad in cancer. You have, a brake and you have a gas pedal. You can have changes like when the brake, which is supposed to stop the car or stop the cell from becoming, 1 to 2 cell to 4 cell to 8 cell. These brakes, you lose the brakes. If you have that Ferrari and you lose the brakes, you end up getting into an accident. Okay, but the other issue is the oncogene is something which, it's like a gas pedal. If the gas pedal is stuck and is continually driving this car super fast, that car is also going to get into an accident. Okay. And there's a lot of genetic changes that can happen in a cell, okay? And they're not all the same. It's not all like this. So if you have a pack of, 52 cards, and I take one of these aces and I change it to one of my favorite games, Uno, you put in this wild card, that would be what's called a mutation, or really one change occurs. But there are other ways to describe other genetic events, not just a card changes from one card, an ace, to an Uno card. You have things that change what we call copy number. This is this is my dog in there. We're playing poker, and he decides to slip in another ace. Okay, so instead of having a card with 52 cards in the deck, now you have an extra ace in there, so it changes the number of aces, or it changes what we call the copy number, and that could potentially be bad. You have extra genes that are the normal amount is regulating, and you increase that number, and maybe the cells will adapt in a bad way. Another way is actually silencing. You have a remote, you could put the, TV, the news comes on, you hear that there's some bad event, you just put it on mute. You don't want to hear anything. Well, the same thing, the gene actually, there is actually a start of the gene, which you can have changes and it can mute the gene. That's called hypermethylation. So important genes can be muted and they can be turned off, just like your remote control. So not all mutations have the same significance. I'm an East Coast guy, I'm a New Yorker, but in LA, everyone loves the Lakers, and not all tall Lakers are the same. There's different skill sets, there's different changes that occur. Anthony Davis is coming, everyone's excited. He's tall, he's good, but is he the same thing as, Kareem Abdul-Jabbar? I don't know. But not all mutations have the same functional significance, and basically there are some changes that can happen. Okay, there's some genes where if you're a Spider-Man guy, he was a normal guy, he got bit by a radioactive spider, and then he became Spider-Man. He actually gained function, okay? And there are a lot of changes that can actually lead to the cell acquiring some new changes, okay? We gained function. But there are also changes, just like the loss of I told you, the brake. You can actually hear Superman, who's actually he's an active gene, and then a change happens, kryptonite is added to him, and he loses his kind of function. So depending on what genetic alteration is and what gene, there's going to be different patterns of what happens. Some genes are going to be turned off. They're going to be what we call loss of function. Other genes are going to be turned on, and we're going to call that really gain of function or oncogenic. Now these genes these genetic changes, everyone has genetic changes, and some of them are acquired during life, okay? And let's see what this is here. Okay, so the somatic ones, you're born, you're an embryo, and then later in life you have a genetic change that can happen. It can happen during division, like you can have a patch of cells, or you can have cancer develop. But there are other genetic changes that happen that could be, either in the sperm or the egg, either acquired during the process of developing into a sperm or egg, or just passed from the mom or dad. And then every cell in the patient's body may have one of these genetic changes. Okay, so we call that, if a patient is every cell in their body which was inherited from, either the sperm or the egg, had an alteration, that we call that in someone's germline change. Okay, that is basically every cell in the body which was inherited from mom and dad has a genetic change. But then the somatic ones are the changes that occur as the cells have already, your organism, your human, they're genetic changes that can be acquired during one's life from some type of damage. Damage could be due to some chemical exposure, or it could just be due to just random genetic change. It could be due to a lot of different things. And now, a lot of these changes, again, you have a normal cell, and the cell can get a genetic event, and a couple genetic events, and a couple other genetic changes, and you have enough of these bad genetic changes happen, and then the cell is basically rolling downhill, that's the Ferrari going at 1,000 miles an hour, going to get into an accident. That first genetic change, it could be something that happens due, you go to a tanning salon, you get radiation, you get a bad event. But it also could be one that you may have inherited from your mom or dad that was basically you had no control over, okay? And we call that basically the germline or inherited change. Now, if you have enough genetic changes, it's not all just I have one genetic change. There's usually many genetic changes that occur. There's a famous geneticist who says you need to have really 7 critical events for a cell to go from a normal function to really get out of control to become a cancer cell. And these need to be coordinated things. Some things that are preventing, preventing the cell from growing. Other one basically avoiding the immune system. Other events that cause the cell to basically be able to learn to migrate and move. So you need to have a lot of coordinated. It's a lot of bad luck in a lot of different pathways for a normal cell to go awry and lead to a cell becoming a cancer cell. So those are kind of some of our glossary of terms, and we'll talk about how this relates to, germline or the alterations in some of these inherited genes. So really, what causes kidney cancer? So we tell, patient comes to our office, we sit down with them, every once in a while, why did this happen, okay? So we know some of the risk factors, we know some of the things that are associated with kidney cancer. This doesn't mean it caused it, but we know things like male gender. Males get it twice as common as females. Increasing age. Kidney cancer is something, or renal cell carcinoma, we don't see it in individuals that are age, like age 2 or 3. The median age of onset of kidney cancer is age 64 with some obviously wide variability. Hypertension, there's slight association. Obesity, there's association. Race, we know that certain races such as African American ethnicity may be a higher risk. Chronic kidney disease, there's a lot of inflammation that can occur in someone's kidney, and that could also lead to maybe some genetic changes leading to cancer. And then there's some rare chemicals that can be associated with kidney cancer. The most common one, which is really well understood, is called trichloroethylene. This was a chemical which was used in the kind of the organic solvent industry. People would go into vats and they clean the vats by scrubbing the insides, or really the aeronautic industry. But a lot of folks who say it's bad luck, okay? And really, what does bad luck mean? Well, it could be a gene which has been altered by random chance. It could be a gene which got altered just due to living, or something that just happened. Or you could have inherited a gene that was passed from your family that may have just been that first. Again, if you need multiple genetic events from a normal cell to become cancer, there's no reason why that first event could not have been a gene which was altered and would have been one step down the road of developing a tumor. So we have gone a long way in our understanding of what of all these basically predisposing syndromes. It used to be we knew only a couple genes which, when they were altered and you inherited them from mom or dad, you would have a higher risk of developing cancer. Now we know there's about 70 or 80 of these predisposing, we call them syndromes, where really they're just, you inherit a bad gene from mom and dad, and you have a higher risk of developing something. So everyone's more familiar with these genes, BRCA1 or BRCA2. They're breast cancer genes, but they are actually genes which, instead of just saying, oh, I have a lot of breast cancer in my family, it was one of the first kind of hereditary cancer syndromes where they understood that if you had an alteration in that gene and it got passed to a daughter or a son, you had a higher risk of developing certain types of cancers. But now we know there's about 70 of these or 80 of these now, and we're just developing, we're learning about new ones every single week. This past week, there's a new pancreatic cancer gene. So instead of just saying, oh, it's bad luck, I have this running in my family, now we're able to put kind of more information and study what's going on in families, and we can identify that there's probably a lot more of these than we kind of knew. And this cannot just mean I have a higher risk of developing kidney cancer. It may be I have a higher risk of developing multiple types of cancers. So really, this is where we are now. We really understand, really the tip of the iceberg, that there are a lot of syndromes, about 15 syndromes that are relevant in kidney cancer, but there's probably a whole host of other things that are polymorphisms related, like that giraffe, they may have different spots, or John being handsome, me being less handsome, that may be also predisposing us to developing a higher risk of developing certain cancers. And there's also obviously a lot of patients who are being failed to be recognized that actually have some of these known conditions, because sometimes it's kind of challenging to understand some of these complex genetic conditions. Now, why is it important to kind of understand which tumors are potentially linked to a genetic predisposition? Well, for an individual, so if we know about 5% of our kidney cancers may be related to a strong genetic predisposition, why would this be important for us to know? So for someone who may have a kidney cancer linked to a genetic condition, well, we want to have the potential to maybe screen other organs. So we've had patients who, they could have another tumor that forms, let's say, in their eye. You may want to screen their eye because you want to prevent that tumor from becoming out of control and becoming a problem. You potentially can adopt some we call well-defined management strategies that have really been studied in some of these kind of rare forms of kidney cancers. You may have some ability to select medical therapy. UW here has had amazing prostate cancer program, and the team here has really led the way in understanding inherited risk of prostate cancer. And then there's actually genes which, if they're inherited and you understand those alterations, you can select for a different type of therapy for these kind of less common forms of prostate cancer. Same thing is probably true in some forms of kidney cancer. This is probably only maybe useful for maybe 1 to 2% of our kidney tumors, but the ability to really select appropriate therapy may be essential in those rare types. And then obviously family members. If someone has a tumor and they have an inherited risk and that is related to a gene, that gene could be passed to a child or maybe linked to other family members, and you may want to start screening or evaluating family members who may have a predisposition. It's not that someone inherits cancer, it's they inherit a predisposition. And I always try to tell people, where do big problems come from? They start out as small problems that really aren't kind of managed early. So this is why really important to know. But unfortunately, the idea of hereditary kidney cancers or really a strong genetic influence has really not been very upfront in our community. And this has gained a lot of traction in other fields. Like breast cancer, there's something called the Angelina Jolie effect, where basically she came out, she really made it known that she had a genetic predisposition to breast cancer. She had a mastectomy. And the rates of genetic testing, the rates of mastectomy actually went way up. A lot of that is just through publicity and understanding, and it became something that it was not like a hidden and people were embarrassed to talk about. It was, no, now this should become first and foremost discussed in the breast cancer community. In the kidney cancer field and other types of fields where it's maybe less common, still there needs to be recognition of these kind of genes can contribute to kidney cancer risk. So how common is hereditary kidney cancer? Well, we're not really sure, but we do think about 2 to 8%. So it's not everyone, but about 2 to 8% of individuals probably have a gene which was strongly linked to the development of their kidney cancer. There is a strong inheritance pattern, and there's 15 known conditions right now that are 15 alterations that if you inherit those genes, you have a higher risk of developing kidney cancer. That doesn't mean that these rare genes may be only 1 or 2 or 3%. The other 4 or 5% are probably genes that we even haven't recognized yet, or really linked to these small variations that may contribute to that giraffe having black spots versus brown spots. And we're learning more and more about it, and we really don't understand things as well as we should. We do know that, like I could tell you, I personally don't know my second cousins, but if you look in registries from like Iceland or Scandinavia, they can trace their family tree back to the 13th century. And you can actually look at about 80% of kidney cancer clusters, or it occurs in 1/4 of the different lineages. So there's a lot of genetic predisposition that if you look back to your 8th cousin, you might have kidney cancers on that side of the family. And it may not be linked to this one gene which we can test for, but there's probably a genetic predisposition which we really haven't fully elucidated yet. Now, we do know, again, probably about 1 to 2 or maybe 3% of kidney cancers are really linked to a strong gene. This would be like if your family member gave you a car and the car really had a broken engine. That car would have trouble driving. Or you inherited a gene which kind of was there was no steering wheel. You couldn't drive the car. So we can test right now for these genes which are like a broken steering wheel or inheriting a no engine. There's a lot of things which I just discussed before, which are those minor variations, which is probably like a seatbelt that doesn't work, a muffler which has like a clink clang in there, or maybe the windshield wiper is not going back and forth. We can't test for those minor changes, but these major changes we can test for, and they're really associated with a bunch of hereditary kind of conditions where patients may have predisposition to get multiple types of tumors. And this is probably the minority of kidney cancer, but if we don't look for them, we don't evaluate patients, we're going to miss these rare kind of cases which have an opportunity to then screen other family members. And these have complex names. There are different genes that are associated with them, and there are different types of things that go on. And we'll talk about these types of rare syndromes. They're very variable. Okay, we have some of our kidney cancer patients who may have a genetic form, and some of these conditions, we call them syndromes, some of them may be associated with a very high chance of kidney cancer. We have a condition called hereditary papillary, which 100% of individuals will get kidney cancer. We have other conditions which can predispose to kidney cancer, where maybe only 2 or 5% lifetime risk of getting kidney cancer. Well, a lifetime risk in a man is about 2%. The lifetime risk in a woman is 1%. That 2 to 5% is not that much higher. So you can go multiple generations and someone will get a kidney cancer and they say, I don't have kidney cancer in my family, but that doesn't mean it actually excludes the possibility that there was a genetic predisposition. And then other organ involvement. We have some conditions that are only associated with kidney cancer, but we have other conditions that are associated with many, many types of cancers. So this is just an example. This is a family that I saw that unfortunately I took care of both these children of this mother, and she had skin lesions called leiomyomas, and both their kids developed kidney cancer, and they both passed away from advanced kidney cancer. But then I see other families that have these skin bumps, and they have no kidney cancer at all in their family. So there's different things called penetrance, is how common you're going to see that. And again, there's some conditions which everyone gets kidney cancer, and there's some conditions which very few people get kidney cancer, and it depends on the gene which is involved. Okay. Now, again, I told you that there's some conditions which get multiple different types of tumors. A condition that we kind of describe the association with kidney cancer is what's called Codons P10 Syndrome. These patients get breast cancer, they get endometrial cancer, colon cancers. So instead of just saying, oh, it's bad luck, I got a lot of cancer in my family, these types of conditions have been associated with a predisposition that leads to kidney cancer. Only 5 to 10% of people get kidney cancer with this condition, but you can put all these constellations of things together, and lo and behold, you can make the diagnosis that someone may have a risk for this, and they can get tested. The other thing is this other condition which I see a lot of called von Hippel-Lindau. Patients, they don't just get kidney cancer, they get multiple different types of cancers or tumors or benign things like cysts in their pancreas. They can get a cyst in their head of their epididymis, but they also can get brain lesions or eye lesions as well. Again, some syndromes, only kidney cancer, other ones going to be multiple different types. There's also different variability in presentation. Some patients are going to get hundreds of tumors. Other folks with these conditions may only get 1 or 2 tumors. It's going to be very variable. Some are going to have tumors that are associated with cysts. Other are going to be tumors that are not associated with cysts. There are some of these genetic conditions that are occurring only with one type of histology. There's some ones that get only clear cell kidney cancer. But there are other types of conditions which patients can get papillary, chromophobe, clear cells. So it's going to be very variable depending on which type of genetic condition. So again, just example of multifocality. This is a patient who took care of that has, obviously these kidneys are very busy. There's a lot of these spots through all this normal area which is bright is a normal kidney, and all that other stuff is all these kind of lesions throughout a kidney. It's obviously a little different than someone who has a solitary kidney. And then operatively, this obviously does not even look like a kidney anymore. It looks like a bag of cysts with a lot of tumors. So again, there's different variability on these genetic conditions. So these conditions are rare, but why do doctors have a really hard time identifying these? And I've seen a lot of patients for second opinions, and not a lot of doctors don't take a really complete family history. They kind of may miss that maybe the mother or father also had kidney cancer. Patients sometimes are adopted. They may not know their family history. Or there's some, like my family, I don't know anyone on my dad's side because there was a bad present given at some party, and my grandmother didn't talk to that side of the family because they were offended. So there's just people don't have a complete family history. And then again, there's difference of what we call variable penetrance, that potentially, a gene may increase your risk of developing a cancer maybe only by double or triple. okay, not like 100-fold. And then there's I have two patients that I've seen the past couple weeks that have a condition called VHL. They have kidney cancer, but their mom and dad didn't have kidney cancer. And this patient, when the mom and dad, the sperm and the egg came together and they had an embryo, there was a genetic event there, and the child developed the condition where they have multiple different types of tumors. So we call that a de novo, or basically a first in family, okay? That someone, just like everyone's familiar with the kind of achondroplasia, or we call it dwarfism, where, a mom and dad would not have, they would be normal height, and then their child would have a genetic change, and the child would have short stature. Well, the same thing can happen with these types of cancer syndromes as well. And then when we evaluate patients, we kind of, have to have a close understanding that, we need to take a complete family history. We need to also do a complete physical exam. I would tell you that, I've been in a lot of urology practices that I'm the only one walking around with a stethoscope, okay? You don't probably see that in medical oncology, a doctor without a stethoscope. So really, and I would tell you, I look at all my patients' skin because, there's multiple types of these genetic conditions which actually patients get lesions on their skin. And we joke around that you need to be an amateur dermatologist to really kind of see some of these conditions. And we work very closely with multiple different types of doctors, pulmonologists, neurologists, to try to evaluate patients who have something fishy going on that you can't really put a hand on it. So, this is a type where if you have a great comprehensive genetics program, which they have here, you potentially have a patient who can get really a full evaluation to look for some of these kind of conditions, because they're actually not that rare. So we came up with a couple years ago kind of a quick and dirty way to really try to help a clinician who may not have seen some of these rare conditions start to kind of put in their mind that maybe that the patient could have a hereditary predisposition. So again, kidney cancer, this is just looking at the distribution of kidney cancer. It's a bell-shaped curve, okay? If you look at height, there are going to be some people who are 7'2". They may play for the Lakers. There's going to be people who are going to be 4'6", who probably may be gymnasts. Okay, but if you look at the distribution of kidney cancer, the median age is 64. When you start going down to someone who's, let's say, 24, that's going to be a little unusual. That's going to be several standard deviations less. And at the NIH, there was a large cohort of patients who've been identified to have a genetic predisposition. And if you look at those patients, their age is clearly very different. The median age of someone with the genetic form of kidney cancer is age 37. That's 25 plus years earlier than the median or average in the general population. So if you have a young patient, that's the light bulb should kind of come on in someone's head. It doesn't mean you have a hereditary form of kidney cancer, but if you have a 22-year-old who has a kidney tumor, you got to say, well, maybe we need kind of further evaluation. So the guidelines of who should be evaluated, who should be tested, is not as clear. So basically, this is our AUA guidelines. It says someone who's young, someone who has maybe bilateral, meaning both kidneys are involved, or multifocal, multiple spots, or multiple other types of cancers. You want to kind of raise red flags that someone needs kind of a further evaluation. It's probably not, most of our patients are going to have kidney cancer. They're going to be somewhere between probably 50 to 75 range. They're going to have one tumor. But if you see enough of these patients that are young, multiple tumors or multiple cancers, you really, if you don't check or evaluate or even consider, you're not going to find those who may have the potentially genetic predisposition. So there's some kind of guidelines that we have suggested, but these are not kind of routine, and not every center follows. Everyone has their own kind of way of evaluating patients, but bilateral, multifocal. The issue is we're not probably screening or evaluating as many patients who should be. For breast cancer, there are clear, strong guidelines. Who should go see a genetic counselor or a geneticist, or who should really have evaluation. But basically what genetic counseling is basically you get evaluated. You have basically what we call really pre-test discussion, going through a family tree, mapping out a pedigree, discussing the implications of testing, then getting blood testing. I do a lot of this myself. I spent 10 years with genetic counselors. I know the whole spiel. I understand do it, but I would say most folks would, a genetic counselor would do this, or someone who is a geneticist. And then patients get tested. Their blood or urine, blood or saliva is tested, and then they get the results back. This is kind of the workflow of kind of the program that we are developing at UCLA. It's a little bit different than some other programs, but basically patients come in, they get a genetic counseling evaluation, blood test, genetic testing, And if they're negative, we have a coordinator kind of disclose those results, meaning we don't find anything in your genetic evaluation. Or if you end up having something that is concerning as a predisposing risk, we get you kind of plugged in the system to get really a comprehensive evaluation. Now, again, I told you we don't have any strong guidelines. In a month, we're going to be having a consensus conference, going to be in Philadelphia, piggybacking on another meeting, and my friend Gennady Bratlovsky, who's another guy involved in hereditary kidney cancer, we're going to have really a consensus conference around really kind of come up with stronger guidelines of really who should be evaluated. And our colleague from MD Anderson will be on our panel, Eric Yonash. He's actually head of the NCCN kidney guidelines. We hope to have adopted kind of guidelines and really who should get a further closer evaluation, because we hear things around the country where there's a 22-year-old person to get kidney cancer and their insurance denies them getting tested because what does the guidelines say? Well, there's no guidelines. So we're trying to make this more straightforward like our prostate or breast cancer colleagues. So I'm going to talk, and the next shift is really the more relevant to most patients who have kidney cancer, some of these acquired genetic changes that occur. Again, these are genes that are changed not from an inherited predisposition. We've just discussed all these conditions, syndromes due to inherited predisposition, a genetic change that comes from mom or dad versus the ones that are kind of acquired during the process of one's life. So again, the cell that gets altered in kidney cancer, renal cell carcinoma, the functional unit of kidney cancer is really the nephron. This is the workhorse of the kidney. Everyone has about 1.5 to 2 million nephrons they're born with. And these basically take the blood, they concentrate urine, and they put the urine into the collecting duct of the kidney, and then you pass it through your bladder out of your body. This cell can become altered, and during life, you can have a lot of genetic events that happen. You can acquire, obviously, kidney disease due to high blood pressure. You can have high cholesterol. You can have smoking. A lot of these damaging agents, or basically just inborn errors that happen, there's damage to the kidney. Some damage leads to the cell becoming killed. Others damage could be the cells acquire genetic events or change as one ages. Now again, if you look at it like you were, it would be the normal nephron you inherited, it was healthy, you get a bunch of mutations that can happen. Some will lead to, not anything that will happen to the cell. Others will lead to the cell, again, just saying, hey, instead of just being a single cell and just doing my job, I'm going to acquire new changes, and I'm going to decide to make buddies. I'm going to go from 1 cell to 2 cells to 4 cells, and then you have, a cancer. But not all cancers are the same, okay? We know that there's some forms of kidney cancer which probably a patient could go their whole life and never need treatment. We have other forms of kidney cancer which are going to be this really rapid, progressive, fast-growing one. And the problem we have in our field is sometimes some of these early tumors, how do you actually know who doesn't need to be treated and who actually does need kind of more aggressive treatment up front? And how do we evaluate kidney cancer? Well, not much has changed in how we have in our pathologic evaluation. This is the microscope from 1670. Our current microscope is not that much different. So if someone has a kidney tumor, we look at it under the microscope, really it's not that much different. The terms have changed. In the 1950s and 60s, we used to call kidney cancer hypernephroma. You guys may have seen that on other places, or what's called the Grawitz tumor. Now we have all these fancier terms to describe many of these types of kidney cancers. But the problem is, looking at it under the microscope is not really any different than it was in 1670. Okay? Can we do better today? Well, we have fancier microscopes. This is our microscope in the lab. Has my daughter with her little kidney pillow on there. We have better microscopes, but it's still looking under the at the cell level at what we call the shape, the morphology. If we do biopsies, we're looking at a little piece of tissue under the microscope, and yeah, I can tell you the shape. Just like I can tell you that someone is their hair color, their height, but I can't really tell you who they are as a person because they need to understand a deeper complexity. So what's called the TCGA, or the Cancer Genome Atlas, was basically your taxpayer dollars going not to build roads, but to really study cancer. And a couple of years ago, there was what they call the War on Cancer, about $400 million congressional appropriation to study about 30 different types of cancers. And the goal was to really molecularly characterize, get a deeper understanding of cancer, and understanding what was driving these tumors to become cancer. And also looking across cancers, not just saying, well, we're going to focus on one cancer, but looking at patterns that emerge within cancer. And basically, this is going to really also look to see similarities and group tumors together. And it's just an example of us understanding the biology of tumors. This is a guy who comes in and he has found that he has a prostate cancer, but his prostate cancer is very similar to an ovarian cancer. I made this a couple of years ago, and now we know that a lot of prostate cancers are actually linked to these genetic changes which predispose to breast and ovarian cancer, the BRCA genes. So we actually would tell you that cancers, there's sometimes kidney cancer between one flavor of kidney cancer, papillary, versus another kidney cancer, clear cell. Just because they come from the same organ, there may be more similarities to, let's say, a melanoma or to a GI cancer. And understanding these differences are going to be really important as we try to make more intelligent selection of therapy, not a one-size-fits-all just because something came from the same organ. And in other fields like breast cancer, they have multiple different types of triple negative, people have said, or there's other HER2 associated breast cancer. Now, kidney cancer, we're starting to kind of break things down further and further. Now, the TCGA, we were lucky because there's only 30 cancers that were studied, and 3 of them were kidney cancer. Clear cell, papillary, and chromophobe are our 3 most common types of kidney cancer. And the TCGA, or government-sponsored effort, was to characterize these tumors. Government also, paid for a lot of people to do these analyses, be on phone calls, devote time, which is all government-sponsored. And there's also ones that we're looking across these. We call them pan-kidney cancer projects, where we're trying to understand common themes amongst all these. And then there's other initiatives which these kidney cancer samples were also studied, looking at understanding inherited risk, looking at other things as well. So we know that kidney cancer is not one disease. We have obviously multiple different types of cancers within this kidney cancer family, and they have different genetic changes. This whole sentence, some of these changes from a normal cell, you have alterations in the first word or the last word, or the different changes are different, leading a normal cell called the nephron, the normal nephron to become a papillary kidney tumor, or that normal nephron to become a clear cell tumor, or to become a collecting duct kidney cancer. Again, just because they came from the same organ, they're very different disease, and we shouldn't assume that the same therapy works. One size should not fit all. Now again, depending on, this is just showing you, this would be the nephron over here, which is making urine, and again, depending on the pattern genetic events that go down a certain pathway, they lead to potentially becoming a clear cell kidney cancer, a papillary kidney cancer. It all depends on the event. But even within these groups of cancers, like clear cell, not all clear cell kidney cancer is the same. There's a lot of genetic changes that are unique to maybe one person's clear cell tumor, but may be very different than another person's clear cell tumor. And we now know it's not just the one gene everyone has known about in kidney cancer since the 1990s, VHL, which is a gene which is found in about 80% altered in kidney cancer. We have a whole host of other tumors, and we were focusing on one particular region of the chromosome 3, and we were only just focused on VHL, but in reality, there were 4 buddies next door which are associated with clear cell kidney cancer, and these are really important in understanding maybe tumors' behaviors, maybe tumors' response to therapy. But really, we know that there are subgroups within clear cell kidney cancer. Now, how can we use all this fancy information? We spend millions of dollars to understand it, but how can we implement this in clinical practice? Well, we know there are key kidney cancer alterations, and we know that they may affect behavior. We can use this information to understand maybe who should be observed and not have surgery, who should potentially be given adjuvant therapy because they actually are more likely to recur. Who's going to respond to therapy with drug A and who's going to respond to therapy with drug B? There are now hundreds and hundreds of papers really describing potential associations with genetics, but how many of these are actually used clinically? Very few. Okay, and actually implementation science, where Dr. Gore does a lot of this implementation science, trying to understand how we take some discovery from someone's lab and actually put this forward to a patient. And it takes many years to implement something, and there's some barriers which we could talk about. Some of them are cost. But basically, what this can be done in practice is you can have a tumor, like here, it gets biopsied, the DNA is extracted, you get sequencing, and you can have these fancy genomic reports where you can generate all this information about the genetic changes which are acquired in one's tumor. Okay, and you can, and it's not just looking at one gene. In the past, you would do sequencing on one gene. Now you can have like a shotgun panel and do 595 genes. You can look at all at once. And you can also look to see what genes are altered in one's germline or inherited as well. The amazing thing is that the cost of sequencing used to be astronomical. So the Human Genome Project, it was $3 billion and 10 years to sequence one person's genome. And there was a race to do this. There's a guy named Craig Venter who actually did his own genome. But there was a big race between multiple different groups, and it cost billions of dollars, okay, and many, many years. But now we can do, within a day, we can sequence someone's whole exome. There are 28,000 genes that are making something versus their whole entire genome, someone's whole entire genetic code. We can do that in a day, and there was a race a couple years ago. They were going to give a big reward to who can get $1,000 genome. It was called an X Prize. That actually failed to someone to win that award, but now we're there where we can actually sequence something for $1,000, someone's whole genome. The problem is, the sequencing cost is one thing, but there's actually also cost to do something in a clinical lab. You need to pay staff, you need to do quality controls, you need to kind of generate a report. So a lot of these fancy sequencing reports or tests, they're about anywhere from $4,000 to $5,000 a commercial lab will cost to sequence a tumor. That we need better efforts to continue to lower these costs. Again, if it costs $1,000 for a lab to do it, it may be $4,000 from a company because they need to obviously make some money, and they need to actually pay their staff to actually run these kind of tests. For kidney cancer, there's some insurers which routinely reimburse this, but again, it's expensive. So this is a patient of mine at Yale who did sequencing, and he sent me this bill, $9,000. He submitted this to insurance. His insurance had denied it. And he ended up arguing with the hospital. They obviously lowered the costs dramatically, but this is unfortunately can be very expensive. Medicare now covers a sequencing for patients with stage 3 or stage 4 relapsed cancers. I'm annoyed by this because I think this may be more relevant to those with early stage kidney cancer, where this information may be very useful in understanding like who needs treatment or which tumor is likely to kind of recur, rather than potentially those who have a metastatic disease, where we can talk about that we have some challenges. But basically, patients can get their tumor evaluated. But the implementation to clinical practice is a little challenging. We'll discuss that. It's not the magic home run that we hope, that we know the tumor alteration and now we know who's going to recur, or we know the tumor alteration, we know which drug is going to be curative. There's some limitations. So again, one way which we're trying to use this clinically, implementing this to actually patient is, we do know there's now about 15 flavors of kidney cancer, and on a small needle biopsy, we're not able to really reliably have the pathologist tell us which is which. Again, you're using this old, outdated technology, the microscope. This is a patient that I saw at Yale that he had a 6-centimeter tumor in a solitary kidney, and he obviously was facing potentially a very challenging situation, a large tumor. It was going to be a hard partial nephrectomy. He ended up on dialysis. He's 82. We biopsied it. The pathologist said this is a tumor which looks pink. We call it oncocytic. Well, we ended up profiling this tumor. Insurance doesn't pay for it, and we clearly, I couldn't tell him it may be this and maybe this. I was able to reliably tell him this tumor, with 99.9% certainty, has all the genetic characteristics of a benign tumor, and he's now watched this, asymptomatic. Okay, and we can do this different type of fancy algorithm. We do sequencing. We end up looking at the copy number changes. Again, you have 52 cards in the deck. You add 2 aces, now you have 54 cards. You take 3 jacks away, you have a little less. By understanding those patterns of gains and losses, we can actually subtype a tumor and figure out which tumor is which on a small kind of needle biopsy. Now I'm going to ask Dr. Suka to tell us which tumor over here that is clear cell, which one is the VHL-associated clear cell tumor? Okay, so under the microscope, everything looks, I can tell you which 7'2" Laker is going to be Anthony Davis, or who's going to, or who's, Kareem Abdul-Jabbar, who's going to have the sky hook. Well, it's probably not Anthony Davis, but they're all both 7'2", basketball players. So we really, by understanding the genetic evaluation, we really can look under the hood and not just say this is a Ferrari. This is a Ferrari who has a bad engine, a good engine. Now, I'm very interested in trying to understand who needs treatment and who doesn't need treatment. So basically, there are going to be a group of tumors which, when they're diagnosed, some of these 1.5, 2 centimeter tumors are going to be the sharks, other are going to be the guppies, other are going to be the goldfish. And I try to explain to patients that these guppies are probably never needing to be treated. The sharks, you're going to need to be treating them quickly. You don't want to leave it alone because it's going to bite you. And then there's the goldfish, which if you leave it in long enough, you give it enough food, it's going to be a big koi. And you probably don't want to go swimming with that fish in a pool because it can also bite you. Now, really, when you have a new renal mass, I'm interested in trying to understand the genetics of this new tumor to really tell which patient, give more information, because every patient comes in with this new renal mass is something which is going to lead to dissemination and spread. In reality, not every tumor is destined to do that. And what we're trying to do at UCLA is kind of do this more risk stratified approach. We know that majority of these small tumors are probably pretty indolent. So we're doing things like more biopsies, looking under the microscope, which is not great. We have a SPECT CT, this fancy imaging protocol, which may be helpful. Really what we call triphasic imaging, where our radiologists are really good at kind of distinguishing subtypes. And then really this genomic biopsy strategy where we could really tell people what they really have, rather than just saying you have a tumor. They say if all you have is a hammer, everything looks like a nail. Okay, and in reality, sometimes you can put that hammer back in and leave things alone. So we have a protocol we're about to open where we are going to do this. We're going to do genomic profiling on a small biopsy, trying to better risk stratify patients. We're going to give them a report. It's taken like a 3-year odyssey to try to get a company to actually get invested in kidney cancer. Kidney cancer, obviously, I'm passionate about it. We all are passionate. We're here on a Saturday. But for a company to develop an assay, prostate cancer, lung cancer, colon cancer, they reign supreme because there's more patients that need these types of tests. Well, these types of tests are important to the kidney cancer community, but these companies, they see dollars and they see less dollars made from these types of tests in a smaller population. So it takes time to develop these types of assays. So we finally found they got grant funding from a foundation to actually pay to do these types of fancy tests. And we're going to now do a trial where patients will not have to pay $4,000 out of pocket like that gentleman you saw. We're going to actually cover the cost. Biopsy, trying to say if a patient, I'm not sure what I want to do, I want more information. Now I can actually profile them on our protocol. We're going to look at change in decision making. But it's taken multiple years begging to try to get companies involved and finding a foundation willing to support this type of work. Now, the other way genetics can be really useful is after treatment. We all have patients who have had large tumors. Patients who have large tumors that are, we say we got it all. Okay, I hate when doctors go out and say we got it all. Well, we got everything we came in to remove. Okay, we don't have good technology to see a single cell. So we take out tumors like this, on a weekly basis. Patient 1 is going to be all happy. They're going to come in the clinic happy. Patient 2 is going to be all happy. And patient 3 is going to have a recurrence. And we're going to say, well, you told me you got it all. Well, in reality, you get everything you came in to remove surgically, but there could be some cells which have already escaped, okay? Well, there are some tumors which are just genetically programmed to escape, and there are other tumors that weren't genetically programmed to escape, and you may be able to learn that from the genetic profile. For many years, there's been many papers written about understanding which tumors are more likely to recur. Just an example, this is my only Kaplan-Meier curve in there to show people that there are some tumors, if you look at number of patients who are living, some tumors are going to have better outcome, where 80% of people are going to be alive at about 10 years. And there are other tumors which have a genetic change, you're going to be less likely to be alive at 10 years. Well, we understand that, but can we actually now implement that clinically? And it's a challenge. So the last kind of couple minutes are just going to talk about advanced disease. Well, we do know that not all therapy works. These are FDA-approved drugs for multiple indications, like psoriasis, okay, neutropenia. The ones in blue are what really works well for patients. So the majority of our drugs that are FDA approved for things like cholesterol or heartburn, they may not actually work great. They may just work okay or so-so for many patients. This, again, due to heterogeneity. Our goal in cancer is to understand these types of alterations and say, sir, your profile is this, you should get drug A. Ma'am, your profile is this, you should get drug B. Or, sir, your profile is both, you get drug A and B. And then you can minimize toxicity and you can really have more intelligent therapy. And the goal is really to say, well, you have this alteration, I'm going to give you this treatment. The tumor is going to be happy, not going to care. Or this tumor, the tumor is going to die because it has been, if you give, I try to explain to patients like kicking a man when it's down. I want to know if this tumor is down so I can kick it and make some really a headway on it. So the goal is to find which patients are going to do best with therapy, but the issues, most of these genetic changes, they're not druggable. We don't have a gas pedal to turn off. A lot of these changes are basically something's broken and it's harder to fix or gene edit, rather than turn off the switch, which is on. So some of our alterations are linked to outcome, but they're not linked to, let's say, a response to therapy. And then those sometimes may be predicted to respond. They're not going to respond. Other patients are predicted to not respond, and they still respond. That's kind of like this PD-1 kind of expression in kidney cancer. PD-1 or nivolumab or pembrolizumab, those agents may work if patients have this marker or they don't have this marker. And then we have done things with sequencing those patients who are exceptional responders. So we have learned a little bit about maybe which patients may do a little better with these class of drugs called mTOR inhibitors. There's some early data that maybe we have some idea which patients may do a little bit better with PD-L1 or PD-1 inhibitors. There's some data that patients who have papillary tumors that are altered in a certain pathway may work better if they're blocked in that pathway. But the issue with any marker is there's heterogeneity. You stick your hand in that M&M jar, pull out this green M&M, and you may not have a good representative sample of what's going on in the tumor. Okay, you're going to say, oh, all these tumors are M&M, great. Okay, in the last minute, basically understand that tumors evolve, and we understand that a tumor, there could be many different kind of animals living in the jungle, and they may have come from a common ancestor, and understanding just one of them may not give you the whole story. We now have done things like sequence multiple parts of a tumor, and we can come up with a similar ancestral tree, and they may be linked. There might be green M&Ms, red M&Ms, yellow M&Ms. And I would tell you that this is something which is relevant potentially to some patients and not to other. This is a palm tree where basically there's a long stem, and this would be like a small tumor where we're showing that a lot of these small tumors are very homogeneous. But there's going to be some tumors where you biopsy it, and this is my wife kind of, she was hiding somewhere in here at this banyan tree. And this banyan tree, obviously you sample this, you're not going to have a good idea what's going on because the biopsy may be kind of questionable. So it may be less relevant, some of these biomarkers, because of this heterogeneity. Finally, the last slide is that obviously the goal is probably find what part of the tumor is active. The whole idea of a blood-based biopsy or circulating tumor DNA, potentially understanding the actual active part of the cancer, taking the blood, pulling out the DNA or pulling out the cancer cell may have some more future than just randomly biopsying someone's lung lesion, when really that may not be representative of the 3 liver spots which are growing. So in conclusion, cancer genetics is complex. We're learning a lot more. I can't tell you which part of this lecture is wrong, but maybe in a couple of years we can readdress it. We have the ability to test a patient's tumor and their blood to understand maybe what caused the tumor or maybe what's driving the tumor. Some of these are related to genetic predisposing events, and some of these are acquired and may be useful in clinical care, but we need to really push forward how to implement it clinically. Thank you. Questions or no? The time. So any brief questions? Sean said we have a couple minutes for. Yep.
Well, you know now about 15 or so mutations that are prescribing 8% of kidney cancer, more or less, 6 to 8. What do you think is, epigenetics factoring in on those? That's what I think about. You can say someone has smoking leading to kidney cancer.
Yeah, we do know that a lot of patients they inherit a genetic predisposition. But they inherit one bad, event. Again, to get cancer, you need multiple genetic events. So things like smoking, things like obesity, there are things that turn on or turn off genes, which we call epigenetic. And there's a lot of interplay between someone's nature, their genes, and nurture, how they develop. So again, there are many ways to get multiple different alterations, but the ways that genes are turned on or off, or epigenetics, is obviously very important. Not understood in somewhat, but the pattern of who gets what and how they influence cancer risk is difficult.
Thank you so much, Brian.
Thanks,
We'll have to have you come back in a couple years to educate us on how we can now actually apply some of your precision medicine concepts in kidney cancer care. Our next speaker is Dr. Sarah Suka. So Dr. Suka has been at the University of Washington in the SCCA for almost exactly a year. We recruited her away from Chicago, and she has a strong interest in how we can eliminate the gestalt that a lot of us have as surgeons and trying to help people prepare to kind of be in their best shape and their best health and with their lowest risk going into major surgeries, including major cancer surgeries like kidney cancer surgery. So we're delighted for Dr. Suka to share some of her knowledge in that area.
Thanks so much, John, and thank you so much to all of you for being here. Brian, that was a phenomenal lecture. Thank you. I always learn something when I hear you talk. So my real interest is in empowering patients to get through, yes? A little shorter. How's that? So my real interest is in learning how we can, as a community of physicians, can empower patients to get through treatment as successfully as possible. And I'm going to spend the next 30 minutes addressing some of what we know, some of what we don't know, and just some things that can help to kind of put you as a patient back in the driver's seat when you're encountering a new diagnosis of kidney cancer. So when someone gets that diagnosis, when someone hears a physician say, "Sir, ma'am, you have cancer," that is potentially a devastating and life-altering moment. And I know a lot of patients have described to me that the rest of what gets said in that 30 to 40-minute interview basically is not heard. You just hear rushing water or silence, and largely because the C word is a terrifying word to hear. No one wants to hear that in their lifetime, and nothing prepares you, even if you kind of had an inkling that something was wrong going on. That's a tough moment, and it's a moment when patients have said to me, "I felt like all control was taken away." And so what we're going to talk about is how you can take some control back, and how we can empower you to kind of what things you can actually manage and handle and alter, as much as a lot of what's going to happen in the coming days to weeks to months after a diagnosis cannot be controlled by you. We're going to talk about what you can actually alter. And so one of the hardest parts is treatment decision making, and there's a lot of treatment decision making in kidney cancer. We're going to talk about some of that. We're going to talk about how you can prepare for treatment, how you can educate yourself, and really improve your chances of having a successful course. So a big thing that I'm passionate about is how to help patients get strong for surgery. So we're going to talk a little bit here about risk factors and how we can understand your personalized risk for tough outcomes after therapy. We're going to talk about how we can improve those risk factors, specifically thinking about things like nutrition and exercise. We're going to talk about habits to kick. How do you take care of yourself as you're going through all of this? How can you help the community of people around you who love you take care of you? And then also how to just kind of manage your own, the emotional impact of this diagnosis. I want to start by talking about nutrition because that's actually one of my main interests. And so nutritional status is something that people talk about a lot. I have good nutrition, I have bad nutrition. We all sort of think we have an idea of what we put into our bodies. And there are some very gross metrics, very blunt metrics that we use as physicians to assess a patient's nutritional status. Probably the most common thing, almost everyone in this room has probably heard of body mass index. Is that show of hands? Everybody knows what that is? It's basically how much you weigh divided by your height in meters squared. It's your weight and your height in a sort of an algorithm. It's what the government and what public policy groups use to define things like underweight versus obesity. I'm going to tell you why it's not so good, and I'm going to tell you how we can get a little bit more information about your actual nutritional status, getting into some more complex measurements. We use lab values. People have probably heard maybe about albumin, which is a protein that we can measure in your blood. We talk to patients. One thing that often happens to patients with cancers is they come in and they say, "Listen, I don't know what's going on. I am eating as much as I can and I just can't stop losing weight." And that's actually something that we know that cancers can kind of ramp up the metabolism in one's body. And so that's actually that's a worrisome sign to us, but it's also an important thing for us to know about because it helps us understand a little bit about what that cancer is potentially doing as it interacts with your body. And then we have more formal nutritional assessments and then body composition assessments, which is actually something that I've gotten very interested in. So let's take a moment, and I'm going to stand on my soapbox and show you a little bit of data and talk to you about how bad BMI is in terms of actually assessing your nutritional status. This was a project that a student and I did in my former job when I was in Chicago, where we looked at a very large patient population, and we looked at their nutritional status using traditional nutritional metrics and tried to understand how those nutritional metrics, those measures of nutrition like BMI, affected things like complications after kidney cancer surgery. And what I want you to take away from this, so this is BMI on the bottom of these bar graphs. The BMI, sort of the WHO, the World Health Organization categories of BMI. And then we looked at patients who had either low albumin, which is considered a low marker of nutrition, versus normal albumins. And what you can see is BMI across, basically, the blue and the red, the gray was the people we didn't have data for, but the blue and the red are essentially, you can have a BMI, you can be severely underweight, a BMI under 18.5, or morbidly obese, class 3 or greater obesity, which is a BMI of greater than 40. And you can have malnutrition at any of those steps. So we may not know just by looking at somebody. I can't say, well, you're overweight, so you're eating enough, so you must be well nourished. No, that's not true. Similarly, one thing I talked about is that severe weight loss. We actually can see that severe weight loss, now here it's a percentage of total weight, so it's a lot harder to lose 10% of potentially, say, 300 pounds as opposed to 10% of somebody starting out at 120 pounds in a short period of time. But massive weight loss can happen at any of these categories of BMI. So when somebody comes in and they say, well, my BMI is this, that's actually a fairly blunt measurement of a patient's nutritional status. And we all know that, like people classically say, if you looked at most of the NFL players, technically most of them meet criteria for obesity. However, a lot of those guys are obviously very fit, and it's because muscle weighs more than fat. So body mass index doesn't tell us anything about what somebody looks like on the inside. So this is where we get kind of interesting. We develop some interesting techniques. So these are CT scans. Anyone who's been diagnosed with kidney cancer has probably had to go through a CAT scan. We have validated measurements that basically allow us to take a single slice from your CT scan, and we can start to measure things like muscle mass and fat mass, and through calculations can actually estimate your total body fat mass and fat-free mass, which gives us a much better idea of what things look like on the inside. And so these are two patients who I took care of actually with bladder cancer, and you can see that their BMIs are basically the same. They're 32 and about 31 and 32, more or less. So on a scale, these people look the same. From the outside, I may not be able to tell the difference. But you can see that what's in red, that's their muscle mass in their mid-body. And you can see that the patient on the right, that patient has a lot of muscle. The patient on the left has very little muscle and actually a fair amount more fat. And that actually has implications for both nutrition as well as functional status and what we think of as a construct called frailty, which is kind of your performance status, which you'll hear a lot about as you go through your assessment for kidney cancer. This is something that I got interested in when I was doing my master's degree, and the concept that I'm going to be talking about is something called sarcopenia, which basically is this concept of severe muscle wasting. Now, this was a term that was defined by a dietitian from Tufts named Erwin Rosenberg. Basically, unfortunately, no matter what we all do, as we all get older, we lose muscle mass. You kind of hit your peak when you're in your 20s and 30s. By the age of 40, everybody's muscle mass starts to decline normally with age. But I'm sure you've all seen all the articles in the New York Times well section, things like that, where they say use it or lose it, right? You can actually mitigate that loss of muscle mass. So even though as we all age, we all lose a little bit of muscle, we all lose our bone density, our metabolic rate goes down a little bit, so it can be a bit easier to gain more fat. Some of these processes are actually accelerated by cancers. And it has to do with the way that the cancer interacts with our body. We don't actually really understand how, and it seems to be something that's fairly variable in terms of whether it's an inflammatory mediator type situation where the cancer causes gross inflammation that causes more muscle mass loss, and then fatigue, loss of activity, and that sort of cycles in on itself, or if it has to do with nutrition, or if it actually has to do with some other proteins that the cancers are putting into our body. But you can see here, this is just an example of a normal 25-year-old's thigh muscle versus a 63-year-old. And you can see that things change with age. Well, cancer, like I said, sort of makes that change happen faster. And so when I was doing my fellowship at the Mayo Clinic, this was the crux of all my research. And we looked at how baseline severe muscle mass wasting influenced outcomes in kidney cancer. And the punchline here, so these Kaplan-Meier curves, these are survival plots. It basically is a way of looking at patients over time. Patients who survive longer will have a higher curve, and patients who are more likely to succumb to their disease will have the lower curve. And essentially, what I'm showing here is that patients who were sarcopenic. So those are these patients who have severe muscle mass wasting, who just had their kidney taken out, not for metastatic disease, but for localized disease. Those patients actually had, after we controlled for all kinds of other variables using some fairly elaborate statistics, there's a 70% increase in their risk of cancer death, and then a 50% increased risk of their overall mortality. So we know that this is actually a very potentially important risk factor that we need to work on understanding the meaning of. It's also been shown to be the similar patterns been shown in metastatic disease in people who had their cancers removed in the setting of having kidney cancer that had already spread. Another problem is that some of the therapies we use actually can accelerate this muscle mass loss further. We're treating your cancer, but unfortunately some of the medicines can make this worse. And that's particularly true with cytotoxic chemotherapy, which is commonly used in other cancers. But some recent data does suggest that even some of our immunotherapies and our systemic therapies that we commonly use in kidney cancer can accelerate this muscle mass loss. So here's where I'm telling you this. So there are things we can't control. One, we can't control the diagnosis. Two, we can't control that some of our therapies are really hard on people. But one of the things that's very interesting about this is that muscle mass loss, that's an actionable target. That's something we can change if we intervene with oftentimes a sort of a multidisciplinary approach using things like nutrition and exercise, and that's what we're going to kind of talk about next. So problem number one is actually recognizing that this is a problem. We can't use BMI. We can't use these old school markers of nutrition, but we can recognize that maybe you're at risk for having low muscle at the diagnosis point, or we can recognize that you're somebody who's at risk. We can watch this over time and see if your muscle mass is staying the same. We can identify you as somebody who's potentially high risk. And then from that, we can basically work on optimizing all the things that we know that can help you keep your muscle mass, help you keep your nutritional status, and ultimately help you keep you strong as you're going through your therapy. So let's turn our attention a little bit to nutritional recommendations. And I think that one of the most common questions I get from patients is, okay, what do I need to eat? The truth is, unfortunately, we don't have a magic pill that will automatically keep your nutrition at the optimal level. And the best advice that I can give you is kind of a common sense approach. We don't need some fancy diet that restricts proteins or restricts one type of sugar, restricts one, don't cut avocados out of your diet yet. There's all these things that are online that can be really kind of confusing. If you start to go consult Dr. Google about the best diet for cancer, there's going to be a lot of things that are going to come up, and honestly, very little of it is based in research. And the truth of the matter is, at this point, we don't really have the perfect diet. But what I can tell you is you need protein. You need and mostly lean protein. So you're wanting to eat things that are generally not heavily processed. Stay away from red meat, but everything in moderation. That doesn't mean you need to cut steaks out of your diet. Fruits and vegetables are important. Carbohydrates are good, but really we want you to stick towards things that are whole grains, more fiber-filled diets, which will also keep you more regular in terms of your bowel movements and help you mitigate some of the potential side effects of some of our therapies. Hydration is key, making sure you're drinking enough water. We're talking about potentially, surgeries for kidney cancer. We're talking about removing the kidney, which is important for your renal function. So we got to keep what kidney mass you have left as healthy as possible, which means you need to stay hydrated. And then good fats, there's nothing wrong with fat. But generally we're talking about staying away from things that are heavily processed, staying away from saturated fats, and sticking towards sort of a monosaturated, polyunsaturated fat diet. So if you look at the basic recommendations that, say, the WHO puts out for nutrition, or we talk about things like the Mediterranean diet, those are kind of good guidelines to go by. So what I tell my patients are, you want to eat the rainbow. You want a colorful plate of food. It's a common sense diet, but everything in moderation, and you don't need to be severely restrictive in any which way. I don't want people going on a diet and not getting their nourishment. Multivitamins are key, but there's nothing, you don't need to be on any fancy supplementation or anything like that. Anytime you're thinking about really changing your diet, though, talk to us, because we can often help you. And then we also have phenomenal nutritionists here at the SCCA who can do a full dietary kind of assessment and give you some advice on where you might make some changes that could help you optimize your nutritional status. What are the benefits of good nutrition as you're going through things like systemic therapy for cancer that has spread, or even as you're getting ready for surgery? You can feel better. If you're well-nourished, well-hydrated, and your bowels are regular and things like that, you will feel better. You maintain your strength and your energy level, you can maintain your weight. I often tell my patients, I don't want you to lose weight, because oftentimes what you're losing is that muscle mass that's really important for you to stay strong. And it can help you maintain better tolerate any side effects of treatment, also potentially lower, there's associations with lowering risks of infection. And after a big operation, it can help you heal and recover faster. I want to take a moment because one thing that patients often talk to me about is also the fact that, the diagnosis of cancer, sometimes we can have that diagnosis, and if a person's just looking at you, they may not know. You may look just fine. You may look totally normal. The people come into my office as patients who are coming to see a urologic oncologist, but they leave as cancer patients. They don't look any different. So other folks may not know that anything is, just from looking at them, anything is different. However, sometimes advanced cancers particularly can really alter a patient's appearance, and that can be actually very devastating and very anxiety-provoking. It can be associated with development of depression. You might see changes in weight, might see changes in muscle mass. Skin changes are fairly common, and patients can internalize that, and sort of every time they look in their mirror, what they see is a cancer patient as opposed to themselves. If this is something that's happening to you, talk to us about it, because it's also something that we can potentially intervene upon, and it's a way of sort of us also understanding your feelings about the tumor and how that's affecting your quality of life. Because one of the things that we work really hard to maintain as we are helping you go through this journey and negotiate treatment decision-making, going through surgery, going through systemic therapy, is maintaining your life. We want you to still be living your life and maintaining your relationships and continuing to do the things that you love to do. So there may be things that we can do to help. And also it's important for us to know if you're noticing things that perhaps we're not picking up in a 15 to 60-minute office visit. Exercise. When I talk to patients about exercise, they usually look at me incredulously and say, wait a second, you just told me I have cancer and now you want me to go to the gym? And I say, yes, actually I do. Because we know very well that your functional status, your strength, your vitality, your energy level, those are all things that if you don't use your muscles as you're going through therapy, you will lose muscle mass. And sometimes just the act of being sort of sidelined, being benched by getting these therapies, sitting in an infusion chair for a period of time, being stuck in bed after surgery, you can lose a fair amount of muscle mass and your strength and your function during that time. So regular exercise during therapy is very important for a number of reasons. It can maintain your muscle mass, it can maintain your performance status, your independence, your ability to take care of yourself and not having to rely on other folks, to do very basic common sort of activities of daily living, like going to the grocery store, getting yourself dressed in the morning. It can avoid the dreaded frailty term, which we have talked about a lot, and frailty is kind of the intersection between your ability to live independently and your strength, and also things like your nutritional status. And then it can also improve how you feel emotionally. I think that's the endorphins, the endorphin rush that people talk about when they actually go out and do something, get outside, be active, do something social. Oftentimes you're with other people when you're exercising, and that can just be good for you. And I know this sounds a little bit like I'm hand waving, but it's actually all very important stuff that can keep you feeling well as you're going through therapy, which we think is incredibly important in terms of how people actually come out on the other side. Now the question is, how do you exercise? And a big part of it is you kind of have to listen to your body. If you were not somebody who ran marathons before your diagnosis, you shouldn't go out and potentially use this as an opportunity to start training for a marathon. You sort of have to work your way up. But you need to listen to your body. I think general recommendations from a number of guideline groups say you should try to do 30 minutes a couple times a week at an intensity that feels comfortable for you. Strength training is really good. Weightlifting actually has been demonstrated not only to maintain, if not improve, muscle mass, but it's also very formative in terms of keeping up your strength so that you can continue your activities of daily living. Endurance sports are good too. We also have a really excellent physical therapy teams here who can help give you individualized recommendations about the types of exercise that you can do that can kind of optimize your function. And also if you are struggling with something, Some patients may notice that they're having a harder time accomplishing some of their activities of daily living that is becoming bothersome. We can kind of help you specifically target sort of getting back on track with those things. Obviously, if you don't feel well, this is not the time to go to the gym, and we want to know about it. And specifically, if you are having problems potentially with parts of your blood counts that may put you at risk for bleeding, things like that, talk to your physician. Let us know. If you have cancer that has spread to the bones, we have to be changing your exercise program specifically. So again, it's really important to communicate with your physician and your care team as you're doing all of this to make sure that we're ensuring that you're doing the kind of work that's maximally going to benefit you, but certainly not going to hurt you. Now, what else can you do? This is a great time to kick habits that have been a problem for a long time. If you are in the moment of diagnosis with any cancer is a very teachable moment. It's a moment that sometimes can give people the strength to get past an addiction or a habit that has been kind of plaguing them that they know they should have quit. So we know that kidney cancer is associated with a number of different causes that, for example, Dr. Shuck just went through. Smoking is a big problem, and we know that smoking is associated in some studies with as high as a doubling of the risk of kidney cancer. So by quitting smoking, not only you can't really change the fact that one has gone from the state of not having kidney cancer to having kidney cancer, but it can also impact how you get through the therapies. And certainly by quitting smoking, we know you decrease your risk of surgical complications, wound healing complications, decrease your risk of infections and pneumonia, and heart attack. Because don't forget, while you're going through your therapy for your kidney cancer, you need to keep the rest of your body healthy. It also decreases the risk of cancer recurrence, death from cancer, and other smoking-related cancers. So this is a great time to talk to your physician about nicotine replacement or strategies to try to quit smoking. Alcohol is another one. We don't know whether or not alcohol directly causes kidney cancer, and there's not a lot of data to suggest that, but we do know that alcohol consumption in high levels is associated with increased risk of complications after surgery. Additionally, your body can develop a tolerance for alcohol that can make the recovery period especially hard because you're not going to be able to be having alcohol, obviously, while you're in the hospital. And so that can really make the recovery that much harder, both emotionally but also physically on your body, and put you at risk for severe complications that can in some situations be life-threatening. So the sooner you quit smoking, the faster you reap the rewards. By quitting within 10 years, you basically get your body back down to the baseline level of a lot of the smoking-related risks. And intensive alcohol and smoking cessation programs have been demonstrated to decrease risk of complications from surgery for cancer by half, which is really exciting. And so it's an opportunity for you to really kind of take control. What else can you do? This is a great time to be rested. Take care of your body. You do need to sleep. You do need to do self-care. This is a chance for you to really take time to take care of yourself. It's also time to let other people in your life know what you need as you're going through therapy, and to really kind of rally the troops to make sure that you're well supported. It's a good time to really focus on making sure you're taking care of all your other medical issues, because if you're having problem with your COPD or with your heart disease or your diabetes as you're going through surgery, that's going to impact your ability to go through the operation. So we really want to help you, and we'll often engage our colleagues in medicine, pulmonary, cardiology, as we're getting people ready for surgery, to make sure that patients are optimized on the day that they are going to be going through their operation, their kidney cancer surgery. And it's really important as you're going through all of this, if you're specifically concerned about one thing, your medications are one thing, but any of these other factors in your life, you talk to your team and let us know about them so that we can help you sort of prepare and develop an action plan, because a lot of times it's just a question of making sure you're seeing the right specialists. We have some exciting work coming down the pipeline here at the SCCA in terms of comprehensive risk stratification. So taking some of these tools that I've talked a little bit about, understanding people's muscle mass, understanding physiologic age versus chronologic age. So not every 80-year-old looks alike. I've met a lot of 80-year-olds who put me to shame when it comes to their exercise programs and have been really inspiring. And so we want to make sure that we're really characterizing what you look like as you're coming into surgery, to give you a forecast of what the recovery will look like that's very personalized. We're working on operationalizing a lot of these more comprehensive assessments in our clinics, specifically looking at how do we understand your burden of comorbidities, mental health issues, nutrition, and something I'm really excited about, which is we partnered recently with the University of Washington Resilience Lab to start measuring things like psychological capital in our cancer patients, because attitude plays a role in all of this. And I know it sounds kind of fluffy, but how a patient reacts when you have that moment of diagnosis, whether somebody says, all right, I'm buckled in, I'm ready to go, let's do this, compared to feeling really alone and scared. Those are, one, it's important for us to recognize how a patient handles that diagnosis. Two, we can help build resilience, we can help build optimism, we can help you get through this and sort of go on this journey in a more successful way. So we're working on measuring those, and then ultimately developing personalized interventions to help patients get through therapy as best we can. We also have a phenomenal integrative medicine team here who focus on bringing naturopathic remedies and sort of using those treatment strategies as you're going through therapy. The other aspect of all of this is, as you're preparing for surgery, this is the time to get all the information you need about what's coming down the pipeline for you. So there's a lot of questions you should have for your physicians and a lot of information that we're gonna give to you. You want to know, there's a lot of decisions that have to be made as one's going through kidney cancer surgery, specifically with respect to, are we going to take out the whole kidney? Are we going to take out part of the kidney? Is the surgical approach going to be minimally invasive or through a big incision? Why is that happening? There are reasons why we do each of these, but there will be some choices that you are offered, and so it's important for you to be fully informed, and that's where we really want to partner with you and make sure that this is shared decision making. This is not going to be, in many cases, there's going to be a lot of input that we're going to receive from you as active participants in your care that's going to help us give you the best possible treatment plan. I'm going to show you a few different aspects of how surgery can impact, how the decision-making impacts surgery. And then one of the big risks, of course, that we're talking about, especially if we are having to remove, in some patients, a whole kidney, in some patients, parts of both kidneys. We're going to talk about renal dysfunction and the fact that in a lot of cases, we're going to have you follow up with nephrology to think about, those are the medical renal doctors, the folks who are going to help protect what kidney mass you have left to make sure that you can maintain your kidney function going forward and minimize, hopefully, the risk of having to use things like dialysis in the future. These are major operations. A partial nephrectomy is the removal of part of the tumor. We're putting the kidney back together. Radical nephrectomy is taking out the entire tumor, often with some of the tissue around it, including things like lymph nodes and maybe the adrenal gland. Cytoreductive nephrectomy, that's a term that we use when we're taking out a kidney cancer in the setting of known metastases. And metastasectomy is actually where we surgically go after the cancer that has spread to other organs, and that often involves us bringing in some of our colleagues who have specialties in other types of surgery. You're gonna have an in-depth discussion with your surgeon prior to going through any of these operations about the risks of the surgery overall, and as I said before, specifically for you. You'll talk about the approach, whether it's an open or minimally invasive approach, and also you'll get a little bit of an idea of what to kind of expect in terms of time in surgery, time in the hospital. Some of these operations are fairly quick, a couple of hours. Some of them are fairly lengthy, 6 or more hours, depending on the complexity of the surgery. In terms of incisions, there's a lot of different options, and depending on what your kidney looks like, what else your body's been through in your lifetime, and the anatomy of the tumor, you may be offered incisions that look like this, that are basically midline incisions or subcostal, sort of larger open approaches. Alternatively, a lot of our work is now being done in a minimally invasive fashion, either laparoscopically or robotically, in which case you may just have a couple of tiny poke holes in an extraction site like you see here in the upper left corner. There's all other things that are different about kidney cancer compared to many other cancers in terms of things that those cancers can do that really impact your surgical treatment. And one of the things that kidney cancer can do is it can get into the vein and spread up towards the heart. That changes things considerably, and so your surgeon's gonna have a very different kind of conversation with you about what to expect and who else is gonna be brought into the team to handle you on that day. This is something that if you're talking about a venous tumor thrombus, we're definitely gonna be giving you some much more specific instructions about the preparation and what recovery will look like afterwards. But I only bring this up to sort of familiarize you with some of the terms that are gonna be thrown about, because I think a lot of the times these can get fairly technical and these conversations can be very difficult to have. But we'll make sure that you're well informed before you go down this road. Finishing up, I want to talk a little bit about recovery and what you can expect. On average, after kidney cancer surgery, you're going to be in the hospital for 1 to 5 days. Surgery, unfortunately, comes with risks, and complications can happen. We'll talk to you about the risks of those complications before surgery, but these things, unfortunately, are part of recovery in some way. We do everything we can to minimize complications and risks, but it's unfortunately a part of surgery. Some patients need additional care and may not be able to get home immediately after surgery, may need to go to a rehab or need extra physical therapy or visiting nurses. We will set that up for you while you're in the hospital. And then we'll talk to you a lot as we start to understand what that tumor looks like in terms of things like the risk of it coming back and what kind of risk stratified surveillance you're going to need. Because unfortunately, we're going to be friends for a long time after this. That it's not just a question of you seeing your surgeon once and then being done with us. We need to see you both in follow-up to make sure you're recovering well, but also to help understand the risk of basically to be watching you to make sure that that cancer doesn't come back. You'll hear words like enhanced recovery after surgery protocols. I bring this up because this is a term that's more commonly used. These are these very careful checklists that we use as physicians in the hospital to basically ensure that your recovery is as smooth as possible. Bringing in expertise from all of our colleagues in nutrition and physical therapy and nursing to get you back on your feet, eating, drinking, and out the door as feeling as good as possible, as quickly as possible. This is an active recovery. We're going to get you back on your feet the day after surgery if we can. We'll get you eating as fast as we can. And so this is a sort of a comprehensive way that we've formalized our expectations for how we get people through the recovery period. And then, again, what can you do in all of this? The act of going through surgery, while you don't get to control much of what's happening under anesthesia, is very much controlled by how patients interact with the staff and the team as they're recovering. We want to partner with you. We want you to have the best experience you can in the hospital. So we're going to ask you to do things to make sure that you're hydrating, sleeping, getting out of bed, eating. This is sort of where we say tough love comes to town after surgery, and basically get you back on deck. We'll tell you what to worry about and how to call us, and you'll know exactly all the numbers to call. We want to make sure that you feel well supported when you leave the hospital after surgery, because those first 6 weeks can be kind of challenging. And it's important to know that it can take a while to recover. It can take anywhere from 6 weeks to feel kind of normal to up to 6 months, and especially if you're going through systemic therapy while you're going through your recovery. Everybody's a bit different. It's normal to have good days and bad days. You may need to change your diet. We may need to get you on new vitamins. But again, we're going to be with you. Your care team here is going to be with you through that recovery and your period of survivorship or moving on to the next phase of your therapy. So I do tell my patients we're going to be friends for life because I don't generally see them about every couple of weeks to every couple of months for years after their operations. And we want you to know that you can always contact us if you have any questions. So ultimately, just to conclude, and I apologize, I've run over by about 5 minutes, as one is going through this experience, this paradigm shift of being diagnosed with cancer to making treatment decisions and preparing for surgery. Your own preparation is key, and your role as an active participant with your care team is critical. We want to help you maintain your nutritional status and your functional status, develop, cultivate, maintain healthy habits, kick bad habits so that we're not doing anything that can potentially cause you problems as you're going through therapy. Really focus on self-care. We want you to come into treatment understanding all of your different options and what to expect. We want you to be in the best shape of your life. I tell patients preparing for surgery is like preparing for a marathon or kind of preparing for war, depending on how you like to approach things. Everybody's got a different sort of battle mindset. And then also understand that this is really a team sport, and we're going to basically develop a team that's going to help you get through this as well as possible. So with that, I'm going to conclude. I want to say thank you for your attention and be happy to take any questions. Yes.
Question, you talked about CT scans and measuring SMI. Is that something that you can measure on existing CT scan, or do you have to add that to the order?
No, so the question was these body composition measurements that I was talking about, do we have to order a special test to do that? And the answer is no. These are just basically measurements that we can access from any CT scan that have been obtained. And so a lot of the work that's been done to date is retrospective. We are working to develop partnerships to basically get these measurements in a prospective fashion so we can actually use that almost as a radiographic lab value. And that's something that's We're working with teams that specialize in artificial intelligence to make those kind of measurements. But that's coming down the pipeline.
One or two more questions?
Anything else? Yes?
I love your part on enabling all of yourself through, you have so little control, and one of them is nutrition. And when I went through the process, I asked, what can I do? And he said, nothing. So my point is, if something is available and you can't, to just say, yes, here, you can do this for yourself through nutrition and exercise. But I really felt like I was left hanging. So is there a process established, a procedure where a patient is given something and it's reviewed with the patient?
Yeah, so I thank you for saying that. And that is what you just described, I think, is one of the biggest problems that I see and I hear from patients, for example, who are referred for second opinions. That moment when one feels like they can't control anything, because there is a lot we can't control. We can't control genetics. We can't control the fact that you've been diagnosed with kidney cancer. But you can control how you handle the diagnosis and how you prepare your body and take care of yourself going through therapy. And I think that act of kind of reclaiming agency in that process is incredibly important. So to answer your question, this is all a work in progress. We're really working hard on developing those educational materials, the interactions that we have with our nutrition service. I actually had a great research meeting yesterday we have a pretty much brand new integrative health care team here who do things like acupuncture and acupressure, massage therapy, nutritional interventions, exercise interventions. So that's something we're working on. But I would say that it's something that maybe as surgeons we've not paid as much attention as we should to in the past years. But again, if you have a question about it, ask us, because there are a lot of things that we can offer you that you can control.
One last question?
Yes, and one of your slides, you listed, I think, avoiding chemicals. You didn't really speak to it. We got chemicals everywhere in our environment. What do you recommend we avoid?
Well, again, I think that as Dr. Shuck mentioned, kidney cancer is a little bit different from some other cancers where there are pretty clear associations between specific kind of chemicals and carcinogenesis, or the development of cancer. But I think that things that you can avoid are avoiding things like smoking and minimizing alcohol. I was talking more on the order of trying to get yourself in the best shape possible for surgery. But for the folks, we have a lot of folks around here who work in industry who are exposed to chemicals, so taking the necessary precautions are obviously important because there may be some risk factors that we haven't identified yet. It's more about just ensuring that you're kind of cognizant of the potential things you're exposing your body to.
Thank you, everybody.
Thank you, everyone.
So we have a short break. Let's try to be back at in like 5 minutes, if we can, for the next section.
Yeah, let's go. Thank you. Hi, I'm Scott Tycody. Many of you I know. I see a lot of familiar faces. But I'm going to kick off the second half of the morning program here. 3 speakers, myself included, until the lunch break. I'm a medical oncologist here at Seattle Cancer Care Alliance. If you don't know who I am, I specialize in seeing kidney cancer patients in our oncology clinic. So we had 2 excellent talks this morning by urology colleagues. Many times the first physician that patients will see with a new diagnosis. But if you're coming to see me, unfortunately, that's usually not good news, that it's a more advanced cancer process, and we're going to be talking about systemic medical therapies for kidney cancer. And so my talk for today, the title, Swing for the Fences, Can We Cure Advanced Kidney Cancer? I wanted to walk you through just a couple of the data points that there is sometimes a conversation in clinic about uniquely good outcomes and potential cure for kidney cancer. We often describe it as a disease that's not curable because we don't want to raise hopes falsely for people. It's not a common outcome. But I think it's an interesting area and I wanted to present that. It's not a talk I've given in this format before. The baseball analogy I use myself sometimes, the idea of hitting the home run for different therapies and different patients. And so I thought it'd be fun just to think about, is that really a reasonable analogy for the likelihood of some of the data suggesting possible cure for kidney cancer?
So I wanted a baseball player, and so I reached back to my own childhood. I grew up in southern Massachusetts, so the Red Sox were my boyhood team. And as I was starting to become a baseball fan, the generational player, the franchise face for the Red Sox was Carl Yastrzemski. You can see here 23 years as a player, all with one team, which in this day and age is really quite unusual. And so just showing his career statistics, total at-bats and total home runs for Yaz, 452. And if you do the math, it means that for any official at-bats, there was a 4% likelihood that you might see him hit a home run. Below that, if you just look across the board, across the major leagues, the last full year statistics, your odds that at any at-bat during a game of seeing a home run is 3%. And if you want to stair right up and really try and maximize your odds of hitting a home run, Barry Bonds, the all-time home run leader for baseball, I'm sure you know, 8% likelihood that in an official at-bat he might hit a home run. So that kind of sets a quantitative playing field, and I think you'll see that is actually a pretty apt likelihood for what we're going to talk about, and perhaps better than, you know, unicorns or leprechauns or something else a little less common. So the language we want to think about, and when you look at some of the medical data, how do we describe a response in patients with cancer in general, including kidney cancer? There's codified response categories as you receive therapy if you're in a formal clinical trial. Progressive disease, stable disease, partial response, and complete response, and those are ubiquitous across the cancer literature. And so just very quickly, progressive disease, you've had more than a 20% increase in tumor findings from your baseline pretreatment scans, or regardless of change in size, you have new sites of cancer. New sites are codified as progressive disease. Stable allows a fair bit of size heterogeneity, anywhere from a 20% increase to 30% decrease in your starting tumor findings is lumped under stable disease. Partial response requires you've had more than a 30% shrinkage in tumor findings from baseline imaging. And again, no new sites of disease. And then a complete response, what we're going to focus on today and talk about, you've lost all radiographic findings of your cancer on scans after therapy applied. And then the duration of that response is also important. If you have a complete response but it's brief and the cancer grows right back, that may not be terribly valuable. So how long does the response phenotype last? And are we or are we not still applying active therapy to maintain that response? The best case being you're done with therapy, you've really gone back to your quote-unquote normal life. So what are the tools that we have to look at? And there isn't certainly time to dive down and look at every single therapy that might be applied for kidney cancer. But just showing you in a table format here, what are the treatments that are available. In 2019, we have 15 FDA-approved drugs for kidney cancer. And we can break them out into a couple of broad categories. There are immune-based therapies. There are older drugs, cytokine therapies, and we're going to talk a little bit about interleukin-2. There's a new generation of immune drugs, and I'm sure some people sitting right here have been treated with these agents, what are called immune checkpoint blocking antibodies, and in the last couple of years there's been new drugs introduced for treating kidney cancer. We're going to look at some of that data with nivolumab, with ipilimumab, and pembrolizumab. And then the other category of treatments, targeted therapies, encompasses quite a large number of different drugs available to us, many of which block signaling through what's called the VEGF pathway, vascular endothelial growth factor, an abnormal growth signal created by one of the gene changes Dr. Shuck was talking to us about earlier today, the von Hippel-Lindau mutation common to the clear cell patients, or another growth factor and differentiation pathway, the mTOR pathway. So a representative drug, sunitinib, or what's called Sutens is the brand name. We'll look at some of that data, and that really is representative of the phenotypes that we see across all the different drugs from this category. To look at these drugs in a different way, to think about the time they're introduced to therapy for kidney cancer, because it tells us what is the possibility to see the long-term behavior of people treated with these drugs. If you look at the timeline at the top, and what I've described as the paradigm changes, common therapy in the field representing new drugs introduced and becoming the dominant drugs for periods of time. So before the mid-1980s and the discovery and the clinical testing of cytokine immunotherapy drugs, There's robust literature about using conventional cytotoxic therapies for treating kidney cancer, but it's brutally disappointing. And there was never a standard of care identified or applied for kidney cancer because nothing worked well enough to really consider it to be beneficial. So the field changed with the introduction of interferon and then interleukin-2. And as I said, we'll look at some of the interleukin-2 data. And that was standard of care, which is really quite novel, a metastatic carcinoma being treated with an immunotherapy approach. So kidney cancer was fairly different than many of the other common cancers. And one of the reasons why it was a fascination to me as in my training being interested in immune-based therapies applied to cancer, and kidney cancer and melanoma were diseases that were quite different than most of the rest of the oncology field. That was status quo for a very long time until a wave of new drugs became available, what we call the targeted therapies. First in class was approval in late 2005 of sorafenib, but rapidly followed by many competitors. And really, sunitinib was the drug that became the dominant medication applied to kidney cancer for a good decade. The world changed again. 2015, FDA approval of nivolumab, first in class of immune checkpoint blocking antibody. And then that once again has really changed the playing field for kidney cancer. Now the checkpoint blocking immunotherapy drugs have become commonplace and are probably the most commonly applied drugs currently for a metastatic diagnosis. So let's start with interleukin-2 and spend a few minutes looking at just some of the data points that guide us in our thinking about application of this drug and really focusing on the complete response profile. It's really the rationale why the drug still occasionally is rolled out today, and we still think about applying it for selected patients. But going back, can you see the mouse? Yeah. The very first data set that led to FDA approval, published in 1995, but FDA approval way back in 1992. 255 patients, many institutions, several independent small studies showing a 14% response rate, but a 5% complete response rate, which was quite provocative in its time. And that phenotype has been repeated over and over again. I show you a couple of large institutions that have published their own single-site experience, the NCI campus where IL-2 was first used as a medical therapy. Our neighbors to the south, Providence Medical Center in Portland, has also published their experience. These centers showing response rates in the 20% rate. But once again, demonstrating that a significant subset of patients are able to achieve a complete loss of all cancer findings undergoing IL-2 therapy. And then a recent attempt to really pool data from a lot of cancer centers and achieve higher total patient numbers to see statistically if you can find interesting associations with outcome. What's called the Proclaim Registry, and our center has participated in that, so some of the data is coming from our own patients. The last report from that registry, 810 kidney cancer patients, published just this year, 2019, 25% response rate, and again, 5% complete response. So across time and across cancer centers, there's this a demonstration that it's a drug that can, in some patients, achieve what we'd all like to have, a complete response of metastatic kidney cancer. And so to graphically represent what happens to these patients over time, this is from the NCI publication a few years back, but a nice graphical display of the point I want to make, which is patients that achieve a complete response of their kidney cancer after IL-2. So at time zero, the graph here on the left side is patients in complete response of their cancer. And the question is, over time, do they maintain a complete response or does the cancer grow back? So if 100% of this population stays in complete response, the line stays flat at the top of the graph. Every time a patient has a disease recurrence, then that proportional amount of patients no longer in response, and the line drops down. So the closer the line gets to the bottom, the worse. It means people are recurring. And so with timeline of follow-up, because IL-2 is such an old drug, you can follow patients for 20 years and beyond. So a 20-year timeline. The take-home message is that the majority of patients with a complete response after IL-2 do not have recurrence of their disease. So 70% of patients beyond 10 years still in complete response. So it's a scenario where we really feel good about using the word cure, that we think those patients probably are cured of their disease and it's never going to come back. The overall survival is a little bit better than the response rate, meaning that if the disease comes back, you certainly have options. You can continue to treat the disease. So very few patients dying of their disease if they have achieved a complete response. Let's look at the same sort of data points if we change track, we look at a different drug. So this is the same sort of analysis with the drug sunitinib or Sutent. So a representative of the targeted therapy class of drugs. And again, I picked out a subset of clinical trials. I could fill out an entire page with a small font with Sutent studies because it's been around now for quite a long time. It was the dominant drug for many years in the field. But if you go back once again to the pivotal data that led to FDA approval, the phase 3 study comparing to interferon alpha, 375 patients that received Sutent, a 31% response rate, but 0 complete responders in that initial cohort. If you move forward in time to the same application, the same drug, compiled early trial results with Sutent, totaling more than 1,000 patients, so a pretty broad representation, 38% response rate, but a 1% complete response profile. The COMPARES trial was a well-publicized trial comparing Sutent to a competitor, pazopanib or Votrient. That study, 500 patients in each arm, a big trial, 24% response rate for Sutent and a 1% complete response rate. And if you jump forward, we're going to look at a couple of data sets with the newer immunotherapy drugs that were tested head-to-head against Sutent as the standard of care at the time the new drugs were coming forward. So this is the comparison arm. These are patients that were randomized to standard of care and got Sutent. Over 400 patients compared to ipilimumab, nivolumab, or compared to pembrolizumab, axitinib. Recent publications, high 20s to low 30s, so right in the ballpark of what Sutent typically shows you in a population of kidney cancer patients, but again, very low complete response profile. So a 0 to 2% complete response rate for Sutent compared to what we saw with interleukin-2. And then once again, if you take that group of patients, so you hone down on the people that had the uniquely good outcome. This is from a European cancer registry where they identified patients that did achieve a complete response with targeted therapies. This was mainly Sutent. It was 100 patients they were able to find. 84 of the 100 had received Sutent. There's a smattering of other targeted drugs. And once again, you set at time 0, the patient has had a complete response, and now you're saying what happens to that patient as you move forward in time. Again, a timeline in years, not as many years as IL-2 because it's a newer drug, but getting out, most of the patients were analyzed for up to about 3 years. And you can see this line falls off and heads towards the bottom of the graph, unfortunately, meaning that with time, patients are not maintaining a complete response, patients are recurring. And this is a fairly straight trajectory. You can see how many patients are analyzed per time point, and so by 4 years, they're basically nobody is being analyzed. But you can imagine in your mind that it's probably telling us that virtually nobody maintains a complete response with Sutent if you wait long enough. And then this was broken out by people, the orange line, people that were had stopped active therapy, and so they had a complete response. Their doctor said, okay, don't need to take the medicine anymore, let's just wait and see what happens, versus patients with the dotted line where we said, well, we don't really know if you're cured or not. If you're tolerating the drug, why don't you just keep at it, keep taking it. Maybe we'll keep things under control. And it didn't seem to matter. The natural history played out with or without the drug being applied. The cancer wanted to come back. So we're not very enthusiastic that the targeted drugs really have a profile, that there's this minority of patients that might have a uniquely good outcome, a complete response that's going to sustain itself over long periods of time. So jumping forward once again, the shiny new drugs that we have that we're using now commonly in the clinic, what are called the immune checkpoint blocking drugs. We're back to the future for kidney cancer. We had cytokine drugs. We were using immunotherapy before it was fashionable. And now we're back to immunotherapy, but now it's no longer a weird therapy applied to a small subset of cancer patients. These are drugs now that are commonplace across the playing field for oncology. It's really changed the face of oncology care, and it's been a dramatic story unfolding. But it's relevant to our diseases. They do work pretty well for kidney cancer. If you look here, the first drug that was FDA approved, nivolumab, again, showing you a couple of the key findings from the pivotal study. 400 patients in this trial got the nivolumab drug versus the comparator. FDA approval and publication of this data in 2015, so it's not very old. We don't have too much follow-on time to see what's happening to these people. There was a 25% response rate with nivolumab, but only a 1% complete response rate when this drug is applied as monotherapy. A competitor for nivolumab, same biology, it has the same target, what's called the PD-1 protein, made by a different company. Pembrolizumab has a study looking at patients that had no prior therapy, The nivolumab data was done in people that did have prior treatment, so not exactly equivalent. A little over 100 patients treated with pembrolizumab. This is data that's just recently been updated this past summer at the big oncology meeting. 36% response rate and a 3% complete response rate. So there are complete responders, but the numbers aren't super high. But jumping down here, really what state of the art in the clinic are combinations of therapy. And so what's come forward, knowing that these drugs are very effective, is asking the question, well, can you start mixing things together and can you improve outcomes? And so these are fairly recent FDA approvals, but these are the things that we're talking with you about in clinic right now as what's considered best available therapy. So a 2 immunotherapy drug combination, nivolumab combined with the second drug in the same drug family, another immune checkpoint antibody. So both immunotherapy drugs, nivolumab and ipilimumab, or Opdivo and Yervoy are the brand names. 550 patients in this study. This was tested again against sunitinib. We just looked at the comparison data a moment ago. So now the immunotherapy drugs, how do they behave? Overall response rate 41%. That's a pretty good number. If you think about the IL-2 data, the Sutent data, it seems like things are getting better with the passage of time. But a very interesting number with this combination, the 11% complete response rate. And that has certainly gotten the attention of the field. I can tell you that that was a popular topic of conversation at this summer's ASCO meeting. A lot of kidney cancer specialists being drawn to this combination by this very high, relatively speaking, profile of perhaps achieving complete response. And one of the newest combinations available just this past April, FDA approval, pembrolizumab and axitinib. Pembrolizumab is an immune therapy. It's a checkpoint blocking antibody, but the combining partner isn't an immune therapy. It's axitinib. Axitinib is the same drug family as sunitinib, targeted therapy. And as we were looking with the sunitinib data, we're not convinced the targeted drugs really bring a lot to the table in terms of the likelihood of a complete tumor response, but maybe when you combine them together, there is some sort of synergistic effect. Overall response numbers for this combination, better once again, 59%. So again, that's a number that's been widely talked about in the field. But complete response rate, 6%. Looks pretty decent, but it's not better than the ipilimumab, nivolumab. But these complete response profiles are beginning to become part of the discussion between doctors, but I think also in the office with doctors and patients. We don't want to oversell the potential that there might be a uniquely good outcome, but it does cross our minds, and we're certainly very enamored with numbers that are moving in a good direction in terms of complete response profile. What's the long-term behavior of complete response with immune checkpoint drugs? Unfortunately, we don't really know. These drugs are too new. There aren't a lot of observations of patients that have been on therapy for many years, so we really can't say. We're asking of the companies that own these drugs, will they not look at these cohorts of patients that have had uniquely good outcomes and show us in some kind of organized presentation at a cancer meeting or in publication, what is the behavior of patients that have complete response? Is it maintained? Are patients recurring? So we can know better how enthusiastic should we be about these outcomes? Should that be a deciding feature of choosing these drugs and which combinations based on the possibility of maybe providing curative therapy for our patients? So just to summarize a couple of different treatment options and what we think about the ability of these drugs to deliver a uniquely good outcome. IL-2 treatments, I would say, because of the very long lead time, represent the best example of the potential to possibly cure a subset of patients with advanced kidney cancer with complete response that's sustained over time with up to 20 plus years of observation and in the absence of ongoing therapy. We didn't talk about how IL-2 is delivered, but it's given only for a fairly brief period of time over the first couple of months of therapy. It has limiting side effects. Patients can't tolerate it for ongoing long duration of time. So it is a drug that does have a very binary point of view. It either works or it doesn't. It works for a few patients in a spectacular way. Unfortunately, it doesn't work for most people, and in this day and age, they move on to some other style of therapy. IL-2, if any of you here have done IL-2 therapy, you can testify that it is not a friendly drug. It's cumbersome, it's toxic. We actually put patients in the hospital into the ICU care suite to safely deliver IL-2 therapy. And because of that, it's really not well aligned with a disease that presents late in life many patients in their 60s and 70s. And so it's really a small subset of young, fit patients where we think about IL-2 therapy because they can tolerate it, versus an older population where that's really not a sensible choice. And so even though there is this unique outcome for patients, we don't offer IL-2 to a very large percent of patients. The newer immuno-oncology drugs are more patient-friendly, so it's available to most patients with advanced kidney cancer. So unique outcomes with that drug class is much more available to most of our patients. The targeted drugs, we think, really don't offer much opportunity for a complete sustained response. And where the field is at is looking at novel combinations. Can we start mixing things together? And can we improve both the overall response profile, but will we start to see in higher and higher percent of patients that might have uniquely good outcome and complete response? So 2 last points in terms of mixing things together. The first is, well, we have IL-2, it's available, we can use it, we have immune checkpoint blocking drugs. Maybe we should use both drugs. What if we give them in succession, in sequence? There is a little bit of data that harkens to that. The original nivolumab study, called the Checkmate 025 study, required patients had a prior targeted treatment in their history, but it allowed prior IL-2, and so a subset of patients had been exposed to IL-2. 42 of the 410 patients that were treated. By overall response, it seemed like the response profile was somewhat diminished in patients exposed to IL-2, but the primary endpoint of the study was really overall survival. And so by that criteria, the IL-2 patients actually did better. You could not quantify the median survival. It hadn't been reached. Not half the patients had died yet, so it's going to be better than 25 months. So in terms of the overall survival metric, maybe there's some additive effect of IL-2. Down here at the bottom, switching things around, looking at an IL-2 treated patient cohort, but realizing that some of the patients enrolled in the PROCLAIM database had a history of prior therapy, and some of that was prior checkpoint blocking antibody. So in this figure here, 748 patients that are kidney cancer patients treated with IL-2, showing overall survival with time, but 17 patients had had a prior history of receiving a checkpoint blocking antibody. And how did they behave? Well, over here, it shows you that even if you had a prior checkpoint blocking antibody, you still could respond to interleukin-2. So there were complete responders, there were partial responders, and the percentage, although the overall numbers are quite tiny, looks pretty consistent with what you would have predicted. The overall survival, again, the red line generally overlaps the blue line. It's much more blocky because it's a small number of patients. And there's a time bias here that works against the red line. The blue line is from diagnosis or from the application of the very first therapy for these patients. The red line, these people already had a treatment applied that's not captured in this graph. And so you would predict the red line would look worse than the blue line, and it looks pretty comparable. So the point being that despite the biology of interleukin-2 and the checkpoint blocking drugs being related, they are both acting on the immune system at a similar target, cells that are called T lymphocytes. We don't see strong evidence for what's called cross-tolerance. Cross-tolerance meaning if one drug doesn't work, that you can predict a related drug that has a similar mechanism won't work, and you shouldn't even bother trying that out in your patients. If one of the immune drugs doesn't work, the other drug class still seems to be valid and still can have activity. So it's rational to think about giving a patient both of these drugs. And so the issue is by sequencing, do you give a patient really 2 turns at bat to try and hit the home run? You give them one drug and you hope that they can have a uniquely good outcome, and if it fails, you can turn around and apply a second drug that also has in the profile the chance of maybe having a uniquely good outcome. So talking about these 2 drug classes begs the question, well, why don't we smash them together? Why don't we give both drugs at the same time into a combination regimen? That idea has certainly been talked about. It's starting to be looked at, and I'm showing you here data from the only study so far that has combined these 2 drug classes. It's a study of IL-2 plus the drug pembrolizumab. This is from Mayor Fishman, a colleague at Moffitt Cancer Center in Tampa. A fairly small study, 26 patients, but this was presented this past February at a cancer meeting, and I'm showing you just some of the outcomes data. In the 26 patients, 20 of the 26 achieved a complete partial response. So the lines here showing you the change in tumor findings. Higher above the 0 line is bad, lower is good. It means tumor shrinking. So 77% response rate, that's the best number of any of the slides or data points I've showed you so far. and 3 of the patients having a complete response. 3 of 26 is a 12% complete response rate. So tiny numbers, you don't want to hang your hat statistically on those findings, but it's trending in the right direction. It looks quite promising. And the duration of tumor response, a figure here that people typically call a spider plot. You're looking at the change in tumor findings for each patient individually as you move through time. But the take-home message at 1 and 2 years of follow-up, the timeline here is in days, you see a consistent trend of maintained anti-tumor response. The lines remain below the 0 line. The shrinkage is sustained over time. So we feel pretty good about the profile of these patients. You really can only see by eye one patient that had their tumor shrink down, but then it's growing back here as if they're resisting the therapy and starting to progress. But that's not the norm. That's really a single outlier for most of the patients having good success. We have a similar trial on our campus, so just an ad for our own program. We have a study that we call by our shorthand the Intruder trial. It's interleukin-2 and once again pembrolizumab, so IL-2 plus a checkpoint blocking antibody. The little Intruder acronym by interleukin-2, the brand name for pembrolizumab Keytruda, and it's for RCC. And so a study that's open and enrolling patients on our own campus. This is a quick schematic for the study. If it's relevant, we're happy to talk to you about it. In the interest of time, I'm just going to skip forward, but we are looking at the same issue on our own campus. So just to summarize, I really wanted to dive down on the concept that we do sometimes see uniquely good outcomes. The incidence of complete response actually corresponds pretty reasonable to the idea of hitting a home run in baseball. It's an exciting event. It's not terribly common, which is perhaps what makes it exciting. We do have patients in our own clinic program that are examples of patients that have had fantastically good outcomes. So we know it's the real deal, but it's only a small group of our patients, and we're hoping with time we can move the bar and have more patients that have achieved complete response. We think that immunotherapy is the drug class that has the property of achieving these uniquely good outcomes, and so certainly our interests in moving forward are looking for novel approaches for patients to receive new immunotherapy combinations and new treatments that might move the bar for our patients. And the last comment wasn't the focus of today's discussion, but we're a big research campus, the Fred Hutchinson in name research faculty, the University of Washington, SCCA is our clinical practice site. But there's a lot happening. I have colleagues that are working on interesting immunotherapy concepts in the laboratory we hope will cross over into the clinic. And I invite you to reach out. It's what we do every day, and we're interested and excited to talk about it. You're not bugging us, and you're more than welcome to come out. If anybody's interested, I can put you in touch with our development team that are happy to lead laboratory tours for patients. I've had many of my patients that have done that and gone to the Hutch campus and looked around at some of my colleagues and what they're doing in the lab. So if that's of interest, by all means reach out, and I'm more than happy to help that happen for you. So I think we started a little bit late. We're a few minutes past the original agenda, but maybe 1 or 2 questions, and then we'll invite Dr. Hall up.
All the charts you showed on partial response and complete response, were those for patients with metastatic disease?
So yes, everything we were looking at was metastatic kidney cancer with whatever drug that we were looking at.
How long can you stay on these checkpoint inhibitors? And if you don't get a response initially, because sometimes it happens later in the treatment schedule?
So yeah, the question was how long can you use the checkpoint drugs, and do responses sometimes happen late in time on these therapies? So 2 excellent questions. The first, there's no maximum duration of therapy. The original development didn't cap the amount of drug that patients could receive. So right now, the orthodox approach is to continue them forever as long as you are judging the patient to benefit. That usually means you've had a partial response, but you still have tumor findings. They stay stable with time, and you just continue forward on the same drugs. There's a lot of discussion in the field about can you safely stop the therapy at some point and not have the cancer findings change. You still have cancer you see on the scan, but it stays stable. We don't know the right answer in that setting, but it's a conversation we often have with people after 18 to 24 months on these therapies. The second question, there's a lot of discussion about the timeliness of seeing anti-cancer effects, and it's quite clear a subset of patients destined to have a positive anti-cancer effect. The onset of tumor shrinkage and disease control is delayed in time. There's no formal guidelines about how long to treat or how long to wait. My personal bias, if you just look, for example, at the sort of spider plot phenotype, you can go back to some of the reports with nivolumab and the other drugs, and you can look at these profiles of individual patients and what happens to their tumor findings over time. And you will see some patients in studies where the tumor findings go up at first, but then they come down. So there was tumor growth and then subsequent shrinkage at a later time. But my feeling is you really don't see meaningful tumor shrinkage beyond about 6 months on these drugs. So on the one hand, I very often ignore an unfavorable first scan, and we continue to go with the same drug, and we wait longer. But I almost never hold patients on therapy more than 6 months. If it's not working at that point, we assume it's never going to work, and we need to move on and try something else. That is a Tychodyism, my own personal bias. There's no product literature or guidelines that instruct you in that regard, but I suspect that's not so different than what other oncologists would propose to you. One more?
The difference, when does partial response ORR change to CR? What is the definition of that?
So I guess the question is, when does a partial response change to a complete response? Very often it doesn't. That's one of the weaknesses of our therapies, is they're imperfect. You see a lot of partial shrinkage of tumor, but then you hit a plateau where everything stays the same sometimes for long intervals, but you fear eventually the remaining cancer becomes resistant and grows back, and ultimately you fail the therapy that's being applied. That is the most common outcome, and that's what we see over and over again. But there are a subset of patients that the rate of tumor shrinkage is very slow, and each time you do a scan, the cancer is a little bit smaller, a little bit smaller, a little bit smaller. If you were within a formal research study and you were quantifying that, you would be a partial response for 12 months, 16 months, 18 months. Eventually, if the cancer shrunk to the point of disappearing, you would finally be a complete response, but only after maybe a year and a half on therapy. I do have an occasional patient that follows that pathway, that it takes a very long time to achieve a uniquely good outcome. So there can be people that evolve from what's labeled a partial response to a complete at very late time points after starting therapy. It's not impossible, but usually you're seeing a predictable rate of change over time with each scan. People that truly have stable findings, that usually is a permanent phenotype. It doesn't change in a favorable way. Unfortunately, it often changes in a negative way over a long enough time of observation. Okay, I'll be around the rest of the afternoon. In the interest of time, let's move forward to our next speaker. So I'm delighted to briefly introduce our next speaker. Dr. Evan Hall is a new faculty member on our campus, a medical oncologist like myself, joining my clinical group with a couple of my colleagues, so seeing kidney cancer patients, skin cancer, including melanoma patients, and coming to us from previously having been at Stanford University and Stanford Hospital, and really just starting in this month. So Evan was very agreeable to come out and speak, just arriving here in the Seattle area. And the title of his talk for us this morning, Improving Quality of Life in Kidney Cancer Patients.
Thank you, Scott, for the warm introduction and the warm introduction to Seattle. This is like my second or third week, so I'm getting settled in. Can you all hear me? Is this, no? Let's see if I can position this better. Is this better? Okay, a little better. I may be too tall for this microphone. How about that? Yeah. Oh, does this work? How about now? No. Better. I'm confused. This is better. Okay, we'll go with this. Oh, this is better. Okay. If at any point you're having a hard time hearing me, just so I can get the feedback, just raise your hand in the back and I can change my positioning. So it's really exciting to be here and to be a part of such an exciting day and to have met several kidney cancer patients and their families. I'm really going to build on what Scott said. I'll say he teed it up for me, so I'll switch from baseball to golf. and talk about, we have so many more choices now for patients with advanced kidney cancer. How do we pick? And one way we might pick might be to use complete response rate, like he discussed at length. But another key thing that often comes up in clinic is, well, how am I going to feel on this drug compared to this other drug that we're talking about? Like, what is my life going to be like? And how we measure that. And that's an area that's increasingly being studied in kidney cancer because we have many more choices. And so we'll talk a little bit about that today and hopefully get some basic information out there about how we think about this. So I have a few disclosures. I've participated in clinical trials of patient-reported outcome instruments. We'll talk about what those are, but they are relevant in this space of how we determine patient quality of life. And I do write and edit content for a variety of patient-facing websites. So I like to start out talks about quality of life with this quote by Oscar Wilde. "To live is the rarest thing in the world. Most people exist, that is all." And I think what we're really talking about with improving quality of life is taking existence, which is very passive. People have talked about feeling a loss of control already today at this meeting, and moving back to a sense of control and empowerment and back to living more fully. And so I think that quote really helps frame our discussion today. So in clinic, when we're talking about what's important to patients with advanced kidney cancer, usually one of the two things at least comes up, and many times it's both, but it's usually some combination of living longer or living better. There are times where it might be neither of these things, but usually these are goals that many patients have. And so we've heard a lot just now about living longer. So these are two of the studies that Dr. Tykoti referenced. The Checkmate 214 study, which looked at a combination of immunotherapies comparing to sunitinib, and the Keynote 426 study, which looked at a immunotherapy, pembrolizumab, with a targeted therapy, axitinib, and comparing that again to sunitinib, which, as we heard, was the standard of care for many years in kidney cancer. And I'll try to use the pointer. Basically, these curves, which we've seen several times today, showed the proportion of people surviving, going from 100% down to 0% over time. And in both cases, the new combination treatment had more patients living for longer than the sunitinib treatment. So that's why these have both become the preferred treatments today. And we're pretty good at measuring living longer. It's, it's fairly easy to classify someone as alive or not alive. So we're able to do that relatively well. When it comes to living better, it gets a little bit harder. And so I'd like to share a patient perspective from a patient I cared for during my fellowship, who engaged in a research project we were doing where we we had a long interview of about an hour talking about his life. And he said he was on chemotherapy for metastatic lung cancer. And he said, so basically I just spend those first 3 days after chemo in bed, and then it takes about 2 weeks to get back. And he was on a chemotherapy regimen where he was treated every 3 weeks. So I said, sounds like only 1 week is really decent. And he said, that's right. And when he came into clinic for his next visit, he was feeling better. He was talking about what he'd been doing over the past few days. And I think there was something that was lost in the communication between the patient and the doctor about what this patient's life was really like during the 3 weeks in between visits. And I think that if the treating doctor had known this, they may have been able to change the regimen to make those first 2 weeks a little bit better, and hopefully the entire 3 weeks between visits better. So it really gets at the core of how we understand what the experience of cancer and the treatments are like for our patients, and how can physicians and patients use this information to make better decisions and improve how people are feeling. So we'll talk about how we are measuring living better. It's a little bit trickier than measuring living longer. We'll talk about what are patient-reported outcomes. Who has filled out a form or some type of survey at their doctor's office asking about how they're doing? Show of hands. Many. You've all filled out a patient-reported outcome. So, and these are being done both in routine cancer care and also often are included in clinical trials. And we'll talk about how we use these instruments with our new therapies. We have new immune therapies, we have new targeted therapies. Can we use the same tools to measure living better, or do we need new tools? So living better, it's more difficult to define. Oh, great, we're gonna get a hook me up. Yeah. Great. All right, is this better? Okay. So we're going to review, I think hopefully if you weren't able to hear anything, you'll still get something out of the rest of the talk. So it's more difficult to define living better. It's subjective, so it can mean different things to different people. It might mean an improvement in physical function, being able to do something that you're not able to do today, tomorrow, next week, next year. Could mean being able to just do more of the basic things to care for yourself on your own. It could be being able to do a favorite long walk or hike that you used to enjoy, being able to train or compete in a marathon. It could be freedom from symptoms that are bothering you, either from the cancer or from the cancer treatments. Things like nausea, vomiting. It could mean improvements in your psychological well-being, less anxiety, less depressive symptoms. And it could mean improvements in social functioning. Maybe you feel well enough that you're able to spend time with your friends, go out to happy hour, go to a Mariners game and sit in one of those uncomfortable seats for 3 hours, but be able to do that relatively comfortably. So when you get into this field of patient-reported outcomes and quality of life, there are a few definitions that come up. One is the health-related quality of life. This is really just an overarching assessment of how someone's health status is as it pertains to their illness and treatment. So basically in the context of cancer, what is the overall health status of the cancer patient as it relates to the cancer itself and also the complications and side effects from treatment? And then the patient-reported outcome is the tool that we use to measure the quality of life. And that's traditionally been a survey, and at the end, maybe during question and answer, we can talk about where that's going. But those that have filled out the paper surveys, that's traditionally how this has been done. And these instruments are designed to look at it in a broad way, so including aspects of physical health, psychological health, mood, finances, on a variety of different scales to help us understand as broadly as we can what the overall quality of someone's life is. So the patient-reported outcome is in contrast to different kinds of outcomes. So I've listed here common examples from kind of different groups, but the patient-reported outcome, the simplest put, is something that the patient completes and no one else interprets it. It's directly from the patient. It's not filtered through someone else. In contrast to the clinician-reported outcome, where a doctor will report or apply a scale after talking to a patient and getting a sense of what's going on. Then patient, family members, and caregivers can offer insights, and those are observer outcomes. And then there's performance outcomes, like how long can you go on a treadmill test, or how do you do on this cognitive test, like an IQ type test. Those are performance tests. And so these things are all different. Patient-reported outcomes in oncology have been used for about 30 years. They were originally kind of conceived of back in the 1980s to evaluate quality of life in cancer patients, usually in the context of clinical trials. And so different survey instruments were developed to measure these aspects of cancer treatment and cancer, including, again, physical, emotional, and social aspects of one's life. And the original instruments are listed here, and these were kind of first brought to fruition in the early 90s, and these are still some of the same instruments that are used in cancer clinical trials today. There are some specific kidney cancer instruments, and I'll just show some of their names because these may be things that you fill out if you're a part of a clinical trial, but there is an acknowledgement that kidney cancer patients have unique symptoms, and so we need to have a special instrument to best describe quality of life among kidney cancer patients. And I've shown on the next slide an example of the DRS, which is really just trying to pull out what symptoms do we think are related to the kidney cancer itself, not even related to kidney cancer treatment. And so this is a survey that, something that you may be asked to fill out. This is a pretty typical looking patient-reported outcome instrument. There's a series of 9 questions, and you're asked to just rate how true each statement is over the past week. I have a lack of energy, I'm losing weight, I've been short of breath, blood in my urine, etc. So things that are fairly common among kidney cancer patients. So you might ask, why do we need this? This seems kind of redundant. I talked to my doctor in clinic. How does this give extra information? I already talk about all this stuff. And that's a good question. And I would say we do need these, and there have been several studies that have shown that when patients and physicians take the exact same information, they interpret it differently. So this is a famous study of lung cancer patients receiving chemotherapy, and they basically, at several different visits, patients and doctors interacted, and then they graded different symptoms that they had. And then they looked to see just how much in agreement they were. And this is the results, and I'll just call your attention first to the top right column. This is looking at fatigue and severe fatigue. And the darker lines are what patients reported, and the lighter lines are what physicians reported. And so after having the same interaction, over time, about twice as many patients said, I have severe fatigue, as the number of patients that doctors felt had severe fatigue. So this is important information that is not always coming through. Again, it's not always filtering through into the clinician's assessment, even though we all have encounters with our patients about how we're doing. So it's adding information. Similarly, in the bottom left, for nausea, the same pattern is true for moderate nausea. We're pretty good agreement on severe nausea here in the bottom right, but about twice as often patients are reporting moderate nausea, which might be at a level that doesn't always get brought up in clinic. There's a lot to go over in a half-an-hour clinic visit, and if it's mild to moderate, they may not get quite as much attention as it should. So these instruments are giving us additional information, and I think that they are valuable. This is one of a large number of studies that have shown this. So how can we use patient-reported outcomes to help us in clinic? There are several ways. So one is in our routine cancer care, and that might help us understand how any given patient is doing over time. We could ask patients to complete these instruments and follow them over time, and it might help us get a sense of what their life is like between visits, if we need to give a bit of a treatment break, adjust doses, try other supportive medications to help their experience or to help troublesome symptoms. So that's one major way that these are being used. The other way is in the context of clinical trials, which, as we pointed out, were kind of their original design, was to help us understand how a new therapy compares to something that we've been using. And so we'll actually look at one of the studies that Dr. Tykody mentioned just to see how these are being used. And then a third way, which we won't talk about much today, is when you have a large group of patients and you can say something about their quality of life, and we have information about how the treatment helps them live longer, we can start to say, what is the value of this treatment, both in terms of helping people live longer but live better? And should this be the standard treatment, or should we look for other treatments? So there is a health economic aspect as well, which we won't talk about much today. So the kind of most famous trial of using these instruments in routine clinical practice came out of Memorial Sloan Kettering. It was a huge study for this sort of supportive care study. They had almost 800 cancer patients that had metastatic or advanced cancer, a variety of different cancer types. And the patients were randomly assigned to either just get their usual care or to have one of these patient-reported outcome surveys given to them electronically during the course of their treatment, not only when they came into clinic, but pushed to them every week when they were at home. And if they reported severe symptoms, the patient's nurse was notified that someone was struggling and they could try to intervene early. And then at each visit, the patient's doctor got a printout of the results of all the surveys that the patients had filled out, and they could use that as a way to start their conversation in clinic. So in shorthand, patients who got this intervention had an improved quality of life. So what we're seeing on the left here is at 6 months after being on the study, the people in blue are the fraction of patients who had an improvement in their quality of life from baseline. And so more patients in the star, this is the group that got the intervention, are in the blue color here. So about 34% had an improvement in quality of life. And this was also true for people who had what was deemed to be a highly significant change in their quality of life. So about twice as many patients who were in this survey group had a significant improvement in their quality of life. And they also needed to use the emergency department somewhat less. So not only can this help patients feel better, maybe it can help use services that no one likes to go to the emergency room. Maybe it can help us prevent some of this unplanned care, which is really distressing for patients and families. They then, this was not planned, but they said, I wonder how the survival was. And they actually showed that there was a small but significant difference in patient survival. So this is actually where living better crosses back over to living longer, and that patients who are doing these surveys and having this extra layer of interaction with their medical teams lived slightly longer than those who didn't. And this was surprising. There's been a lot of effort trying to figure out what is the magic here. You know, is it the whole intervention? Can it just be one piece of it? And there's a lot of discussion still about exactly why this happened, but it is quite provocative. So let's look at the Checkmate 214 study, where the two immunotherapies, ipilimumab and nivolumab, were compared to sunitinib. We've heard about this study and how it's changed frontline care for people with metastatic kidney cancer. And this was based upon largely on living longer. So at 18 months after starting, 75% of patients who got the two drugs were alive, compared to 60% with nivolumab, or with sunitinib, sorry. So that's the living longer. How about the living better? So in this study, patients completed these surveys every 4 weeks. And what they looked at was each patient did it before they started the treatment. That was their baseline. And then they looked over time at their quality of life. And here, higher is better quality of life. Below the dashed line means the quality of life diminished compared to baseline. And what you can see is that, in general, the nivolumab ipilimumab group had a higher quality of life than sunitinib. And that if you compare back to baseline, it seemed like it was a little better than they were before they started on the treatment. It gets a little bit complicated because if you look, there are 400 people that started this questionnaire at the beginning, and then a lot fewer people fill it out as it goes on. So this is a challenge in this field, in that when people aren't doing well, they aren't feeling well, or they come off of the study because the medicines aren't working, we tend not to get their data. So it makes it a little hard. But I think if I'm in clinic and I see this and someone says, I'm trying to decide between these two medicines, I think you can infer that certainly ipilimumab and nivolumab is not worse tolerated for patients than sunitinib. Quality of life seems to be at least as good, maybe better. This line is pretty considerably above. So I think that's useful for the clinic, the encounter in clinic, and provide useful information. So we talked a lot about immunotherapy in the last session with Dr. Tykoti. The instruments that we're using were all, the core of these instruments was designed in the 1980s, which, as we saw from his timeline, predated all of our current immunotherapy strategies. Are these tools still the right tools to measure this? And so a group that I led at Stanford during my fellowship, we looked back at all of the studies of immunotherapy drugs, of checkpoint inhibitor drugs in any cancer, and looked at what we learned about quality of life from those studies. And in general, in all the studies, 15 different studies, different cancers, lung cancer, kidney cancer, head and neck cancer, quality of life was similar or better among patients getting checkpoint inhibitors than whatever it was compared to, whether that was chemotherapy, whether it was a targeted therapy, whether it was placebo. What we also found is that the instruments, having been designed in the chemotherapy era, didn't always ask questions that we thought were the most relevant questions to ask patients, and that we may not be asking people about side effects that we know are common with immune therapies. So if you don't ask, you're not really going to find out. Most of these questionnaires don't have a free response column. It's kind of, you circle the corresponding item. So we know that common chemotherapy symptoms, nausea, vomiting, and fatigue, are really well assessed on all of these instruments. They ask about them repeatedly, sometimes more than once. But that things that people get on immunotherapies and checkpoint inhibitors are not asked very often. Itching, which can be really life-altering, is never asked on any. I have not seen an instrument that asks specifically about itching that's commonly being used today. Rash, fever, muscle aches, things that we hear about all the time in people who are on checkpoint inhibitors in our clinic. These are not commonly asked for on these instruments. And so I think what we need to do is redesign how we're measuring this to make sure that we understand what the patient experience is like on these drugs. And I think this is vitally important, as we move into the new era, where we already saw what we're going to be using today, but 3 or 4 years from now, I hope we're using new and better immunotherapy strategies. But in order to know that something feels better and is better tolerated among patients than what we have now, we have to be sure we can measure it accurately. And so I think that's the challenge that our field's at right now. And there's a lot of excitement about how do we do this, how do we use mobile technology. There was a wonderful presentation at ASCO last year where people, instead of filling out a 30-question survey, they could just pick an emoji that corresponded to how they were feeling. And that actually corresponded pretty well. So there's all sorts of new development that'll make this easier and less laborious for patients while still giving doctors the information that we need to know how patients are doing. So I'm going to wrap it up. I had a great team at Stanford that we worked on this with, and I'm looking forward to continuing this work here at UW and SCCA. I've been very welcomed by everybody here. It's been an awesome first 3 weeks. And I'd like to thank all the patients and their families who've come out today and who participate in this research, because it really doesn't happen without patients and families. So thank you. We have potentially time for a few questions. All right, I see a couple hands. Maybe over back there.
I was just wondering, DBM is a doctor of veterinary medicine.
Yeah.
They study this in animals.
So that was our librarian on the study. When we did that large review, we had a research librarian. They happened to also be a veterinary medicine degree, so she was both. Yeah, read the acknowledgement slide very closely. I think there,
I was just wondering, since these immunotherapies are so new, it is hard to know what to ask about it.
yeah.
And the experience is so really varied, whether people get a lot of pneumonia or diarrhea and all that. So I do encourage you to cast a very wide net.
Yeah, so that's where things are going. So the National Cancer Institute has developed basically a version of what we use on clinical trials to report adverse events that's specifically for patients and that patients will report. It has about 100 items, so it's a little bit long, but it does cast a very broad net. Because I think what's clear is that we find new things that come up all the time. Itching was one of the things that I hear about regularly in clinic that is a little bit unusual, and so we do have to maintain an open mind and not try to make too many assumptions about what symptoms people might have. I agree. In the back.
I just want to ask you one question. It's not specific to anything you're talking about now. But are we going to turn you into a Husky fan, or are you just
So, great question. So I am happy to report I grew up in Shoreline, so I didn't go to the University of Washington, but I always rooted for the Huskies. I never really got on board with the Stanford Cardinals as far as sports, so I will continue to go purple and gold. Yeah. Yeah.
Suggestion to add to your study in this is that when you're going through the treatment process, you come in every week or every 2 weeks, and you usually see the same patient coordinator, and that's the person taking your statistics and knowing you the best. When there's a vacation change or a personnel change, and you're at the critical stage of your treatment, that there's extra precautions taken for a better methodology to know that a patient is in crisis, and that the treatment plan maybe should be altered, but because that person doesn't know you and doesn't have the same exact qualification tools, that it changes the outcome of the patient's care.
I completely agree. I mean, the relationships that we have with our patients and even our nurses and our whole care teams are vitally important, and it's really disruptive when any part of that group changes or when
Everybody needs time off.
it is. It's hard.
Very important.
And I think as it applies specifically to this, it even matters, under what circumstances patients are filling out the survey. If it's the same time and it's right before you're going to see Dr. Tykoti, who you have a great relationship with and always makes you happy, you're in a certain mindset. If it's 8 days since your last dose and you're at home and you're having a hard time getting out of bed and you're filling the survey, that's a whole different place. And so we know that even small differences in how the instruments are delivered and when they're given make huge impacts on what we learn. So I think that's right on so many levels. Right behind you.
Are you using commercial strategies to get higher survey compliance?
So survey compliance is a challenge in this field. Even in the context of a clinical trial where we have a whole team that's designed to ensure that things are following a protocol, the numbers on those usually range between 70 and 80%, and that's where it's been, we have a whole infrastructure to help that. There are apps, I'm not going to name any because I don't have any favorites and I don't want to bias anything, but there are apps on the Apple Store right now that do a lot of this monitoring for you. So there are already commercial products that are aiming to do this. I think there's still a lot of tweaking that needs to go on as to how this is best implemented. How to get this information to your doctor in a timely fashion, all that is a real implementation piece. We've heard a lot about implementation science, and so that's a real challenge for cancer centers to figure out how to do this best. All right, we are out of time. Thank you very much.
Hello. There we go. Thank you, Scott, and also for inviting me. As Dr. Tykody said, I am one of the radiation oncologists practicing at the University of Washington as well as Seattle Cancer Care Alliance. And my specialty is actually brain tumors and brain metastases, but I overlap quite a bit with this group with patients who do have brain metastases from renal cell carcinoma. And I have to say, some of these patients have been one of my favorite parts of my career. I think because over the last 15 years, things have really changed for them. And I have lots of patients who I've treated who have advanced disease, and they have done quite well with all the new systemic therapies. So today, my aims are to discuss a bit about radiation oncology and to talk to you guys about how we look at our field and what your experience is going to be, and then also some of the ways that we are pushing the field forward. So my learning objectives are pretty simple. One, what is radiation oncology? Actually, many physicians don't know what radiation oncology is. We're kind of this small field in the basement. So we don't always, people don't always know our processes. So I think some of you have probably undergone radiation, so you will know this, but some of you probably haven't. So I kind of wanted to talk about that. Second, when do we use radiation therapy for kidney cancer? And we'll kind of go through different scenarios and thoughts. And then finally, are there really up and coming ways for radiation therapy? So first, what is radiation? So our physicists will call radiation energy traveling through space, which is quite a broad definition. We work with physicists quite a bit. But I get this question a lot, and really radiation is under the electromagnetic spectrum, this whole spectrum. But when we talk about radiation we use for medicine, it's right here on this ionizing radiation. So it's here with very high frequencies with gamma rays and X-rays. That's very different from the visible spectrum we have, or really long waves like radio waves. So we're really here with the ionizing radiation. And as far as ionizing radiation, it's actually a very natural treatment. We always kind of laugh about that, but it's true. That most of the radiation that we actually encounter is in the ground, buildings, cosmic rays, food and drink, very low doses that you get. But about 15% are actually artificial sources, and most of that is used for medical use. X-rays and what I will talk about, which is therapy. So how does radiation actually kill cancer cells? It works very much like chemotherapy. It damages DNA. So you see here is an X-ray and ionizes this area here, uses water and hydration and oxygenation to make nicks in the DNA. And then that tells the cancer cell to die over time. So very much like chemotherapy. Our field is pretty old. We started using radiation for cancer in the late 1800s. In 1901 was when the Nobel Prize was awarded for X-rays, to kind of give you an idea. This is Dr. Röntgen. In 1903, there was actually the Nobel Prize in Physics was given to 3 other scientists, including Marie Curie, who's one of the preeminent women scientists from before, for their research looking at radium, which was a radioactive source, and using that for cancer. But over time, radiation has really changed as a field. So we started off, like I said, in the late 1800s, treating patients with X-rays. And then in the 1920s, quite a bit, we were using radium, which is a radiation source, to treat accessible tumors. So if you had a tumor you could see on the skin or anywhere, you could put it right on there, and it would shrink away. But our modern radiation really started in the 1950s, and that's when we started to come up with our modern machines called a linear accelerator. A linear accelerator is what makes the radiation, and it's our modern machine of today. But over the years, in the last 70 years now, we have used a lot of technology and better imaging, as well as some other technological advances in radiation to really make radiation much more pointable at deep tumors, much more precise. And imaging has really changed our field as well. So in the 1960s, we first started with radiosurgery, which I will talk a little bit more about, which was very pinpoint radiation and the ability to pinpoint that in the brain. We then started using a lot of computerized planning and algorithms to really calculate radiation, no longer just sticking it on somebody's skin, but actually calculating how to get it there and being very precise. We then had a lot of advances in terms of the type of radiation and the computer programs that we could use to create these plans. So in the 1990s, we started using something called IMRT, or intensity modulated radiation therapy, that allowed us to make very specific shapes and plans in the body. And then finally, more recently, we've used a lot of new technology like proton therapy to be even more precise. So the idea in looking at radiation oncology is radiation is not a new therapy, but what has really changed is our ability to point it and be precise. So how does a patient actually get radiation? These are kind of the 6 steps that I introduce my new residents to. And I wanted to kind of go through each of these steps. Like I said, some of you may know about this and some of you may not. So the first step, which I think we do quite well at SCCA and UW, and which I love about practicing here, is that we're part of a multidisciplinary team. So we work with the medical oncologists, as well as surgical oncologists or urologists, to see a patient, and often we do this in a multidisciplinary clinic, which is my favorite way because we can kind of talk back and forth to each other. But we really see a patient, review the records, meet and examine the patient, collaborate with our team, and then we discuss patient recommendations. So we're part of the multidisciplinary team. If we decide that you need radiation, then our first step is to do what we call a simulation. This is where we work with a team of radiation therapists. So you'll see that radiation oncology is a field where we have a big team. And though your radiation oncologist is your doctor who you see and runs all of this, you will meet a big team. So this is a few of our therapists here, and they are setting up, well, she's a therapist as well, but she's posing, molds and devices to position a patient. So the idea is if I make a radiation treatment plan, I want to make sure my patient is in the same position each day so that all that precision that I planned actually is delivered correctly. So we do that. We make all these molds. She is sitting or laying down on a blue kind of mold that's custom fit to her. And then we get a CT scan, which is right behind her for planning. Oh, I guess I do have this. Maybe. There we go. The next step, and I apologize a little bit about this image. I realized a little too late that it had gotten not quite on. But the next step is one of my biggest steps, which is when the radiation oncologist then takes that CT scan and contours out where they want it to be. So right here is a tumor coming out of the kidney, and we draw around exactly where we want to target. And this is where we use a lot of different MRIs. We use MRIs before surgery, after surgery. We use PET scans, CTs, many things to really be able to see where the tumor is. So the surgeons can really see when they're in there physically, and we see by imaging. We also have a lot of discussions with surgeons. I think this part is really important about multidisciplinary care. I will talk to surgeons and say, where did you think you left tumor? What do you think it was invading into? So that kind of communication is very important in trying to decide where to deliver the radiation. So the fourth step is treatment planning. So once I give my dosimetrists the contouring or where I want it to be, I tell them, I want you to get a certain dose to this thing, and I want you to avoid the bowel right next to it. And then I actually work with a team of dosimetrists who are trained to use our computer programs to put this all together. So we have kind of differing types of plans. At the one on the top here is very much a basic plan. This is kind of as basic as you get because we come from the back, this green, and from the front, it's red, and you get this blue square of radiation that goes around the pink, which is what we drew out, and just have a very simple plan. You came from both sides and you box the radiation right around. But actually what we've evolved to is much more complicated than that. Below here is a lung tumor where we use kind of multiple beams, so those yellow stripes are all beams coming from different angles, because that allows us to really create a plan where we're very precise on that tumor. So you can see a big difference between here, someone's pelvis, a big part of it getting radiation, and this one where only this little area in here would be getting radiation. So I work with a plan. The dosimetrists put it together as I tell them to, and then they give it to me, and I tell them, tweak this, and we go back and forth, and that's why planning can take several days. The next step is quality assurance. So we work a lot with medical physicists, and a lot of people have never heard of medical physicists, but they're often PhDs or master's physicists. They work in medical physicists, and they help us by checking all these plans, doing quality assurance, and ensuring that we are giving the right dose. So here is one of our physicists checking some of the hardware that we use for delivering the radiation. And finally, treatment. So during treatment's actually given by a team of radiation therapists. These are trained personnel who will set up a patient in their mold and everything that we made. They'll use CT imaging and X-ray imaging to get them in the right position, and then they'll turn on the machine. And so radiation is actually delivered by a team, and it's delivered as designed by me, the radiation oncologist, but they make sure it gets delivered correctly. So those are kind of the 6 steps of how it works. And my next topic would be then, how do we use it in kidney cancer? So you may recognize this slide. I already talked about how radiation will damage cancer cells. And people always ask, which is a good question, well, if you're damaging the cancer cells and you're pointing it right through my abdomen, why are you not damaging what's next to it? And part of that is because with cancer cells are dividing constantly, and so they're constantly going through this division, and so radiation affects them quite a bit. Normal cells are not dividing as quickly or as often, and also can have regeneration from stem cells, so it's able to withstand certain doses of radiation. So traditionally, radiation is given over several weeks, so 25 to 30 treatments over 6 weeks, and you get a little bit each day. And this graph kind of represents that, because what this graph actually shows is that this is sort of the number of cells, because you think about all the millions of cancer cells, and then every day it gets hit and hit and hit and hit and hit over and over again until you get as few cells as possible. So your aim is to get rid of all of the cells of radiation. And so this is our traditional way of giving radiation. And for kidney cancer, it didn't work very well. Did not lead to good control. And we kind of abandoned it for much of kidney cancer. But actually, when there is other ways to give radiation, you can give it at a higher dose, but just in 1 to 5 treatments. So instead of giving it over 30 treatments, just 1 to 5. And we think when you give a higher dose, you have a little bit of a different curve. So if this is the number of cancer cells, you hit it once, it sort of falls off very quickly. And we think of that as more of a ablative dose. So it works a little bit differently than the other way. But the thing about high dose is that high dose requires precise targeting because you couldn't hit a big area with a big dose of radiation without hurting somebody. And so what has allowed us to use kind of higher dose is our technology. So I showed this before, this lung tumor with the multiple beams coming from different areas. And then this is a picture of that lung tumor within the lung. The patient's kind of laying on their back and we get a slice. And you can see that the lines around here, which represent the dose of radiation, I kind of think of it like a mountain. So highest dose here and then it falls off to lower and lower doses, is really quite specific for that tumor. So this is stereotactic radiation therapy. And what is stereotactic radiation therapy? It basically means precise delivery of radiation. And there are different names that you may hear. There's stereotactic radiosurgery, SBRT, which is stereotactic body radiation therapy. Some people came up with SABR because they thought it sounded cool. Stereotactic ablative radiation therapy. And there are many different machines. And if you have done any reading or seen advertisements, you'll see lots of advertisements for different machines. So there are linear accelerators, there's Gamma Knife, which I use, there's CyberKnife, which I've used in the past. And what these really are machines. So these are some pictures. They always like to make very dramatic things happening with radiation, but these are all pictures of different machines that give the same thing, and the idea is very precise radiation. So in terms of kidney cancer, the biggest role that we've had is in metastatic disease. As I said, brain metastases, which I will talk a bit more about and where I interface with kidney cancer patients. Painful bone metastases is another area, and then other metastases. There are some early studies about non-resectable primary kidney tumors as well that is starting to emerge. So brain metastases. Brain metastases from kidney cancer or breast cancer or lung cancer occur because cancer cells will circulate in the blood. And when they circulate in the blood, it circulates up to your brain. And for reasons we don't quite know, a cancer cell may stop and make a tumor. So you can see here in this picture, you have the primary tumor, and it goes through the blood. And then when it gets to the brain, for reasons we don't quite know, there might be one that stops and makes a tumor. And so brain metastases from kidney cancer can happen at different times, but it's more likely to happen. It's not common, but it's more likely to happen for a patient with metastatic disease. And the hard thing about brain metastases, as you can imagine, is that a tumor in the brain may cause symptoms and neurological symptoms that greatly affect someone's quality of life. This is changing, but part of brain metastases issue too was that it was not always treated by the chemotherapy that was given. It was not always treated by the drug therapy. And that's because the brain is really covered by the blood-brain barrier, which protects it from things that we shouldn't have in the brain. But it also keeps often a lot of drugs out. So sometimes people will be responding really well to drug therapy, but will have brain metastases. And that's where radiation really comes in. That is changing, I have to say, with the different therapies now. The immunotherapy Dr. Tykody was talking about, many of the targeted therapies get up to the brain, which is fantastic because we then are seeing fewer and fewer people come back with brain metastases in that way. But here is one of my favorite patients, a patient who has a brain metastasis in the left temporal lobe. You can see here that white. This is a brain MRI, and when you get that contrast, the tumor really takes up that contrast. And this is the Gamma Knife day plan where the high dose is really just this yellow right around it, and then the dose falls off very quickly. This patient actually, if you look at the blue here, that was a previous treatment that patient had had. And this is what I find with my patients with kidney cancer and brain metastases is that I have a whole bunch of patients, and this is why it's one of the most gratifying, where they can go many years without a brain metastasis. And even if it does come back in a few years, I can treat them with radiosurgery. And then they go another few years and I can treat them with radiosurgery and they're feeling great and doing everything they did. So this is where I think it's important to follow patients because with this type of high dose radiation, kidney cancer will respond well. And so it's important to kind of make sure that we get the brain metastases when they're small, because we can really keep people's quality of life and not to have them suffer from it. This patient, 6 months later, that's what it looked like. So it just kind of scarred down. And I tell people this a lot because it's always a little hard to explain. With surgery, it's pretty easy to think about. You take a tumor out, can't see it anymore. With radiation, you cause it to die, but you often still see a little nub in there. And it's just kind of a little bit of scar tissue or cancer cells that had died. So we also get involved sometimes with bone metastases. Metastases of the bone can be painful and can put bones at risk for fracture or breaking. And so we use standard radiation for this. We also use stereotactic high-dose radiation. This is a patient with a stereotactic plan. They had a metastasis in the bone in the spine, and that's a very specific plan. And we also have metastases that pretty much can be anywhere. That's where we can design fields depending on where the metastasis is. This is a patient of mine who I actually treated about 6 years ago, had a very large tumor in the nasal cavity that obviously caused him a lot of symptoms. I loved looking back at this because he's doing so well now, and he sends me his images every year, and it looks great. So I think very important that we use our radiation in terms of getting patients to their next systemic therapy, and just use it in the places where the tumors are causing problems. So finally, just a little bit of word on early studies. There are some early studies also of treating primary kidney tumor, which if the patient doesn't go through surgery. And this is made possible, again, by our stereotactic and our ability to be precise in getting the radiation there. So finally, what are the up-and-coming uses for radiation? So I think, actually, sometimes people ask, are you going to be out of a job because, immunotherapy and everything else is going to take care of it, and you're not going to be able to use your radiation? And I say, I would love to be out of a job. I could certainly do something else. But I also actually think what I've seen is that as our immunotherapy and our drug therapies get better and better, we actually are finding a lot of roles for radiation, which we didn't have before. And one of the roles is with the fact that radiation, which used to be thought of as suppressing the immune system, actually there's more and more data that it really triggers an immune response. So the thought is it works kind of like a vaccine where the the cancer cell dies because of the radiation, and it gives out a lot of tumor proteins and antigens, and then the immune system recognizes that and starts to attack. And so what we see is that radiation does work with immunotherapy. It seems to enhance the immune effect. There also have been described what they call the abscopal effect. The abscopal effect is very interesting. It's not always seen, but what we've seen is that sometimes when you treat a patient for one tumor, another tumor that you did not treat actually may respond. And so the idea is that the treatment with the radiation really kicked up the immune system. So this is a really intriguing event, especially when our patients are on a lot of immunotherapy, because this idea of helping that immune system really attack the cancer is quite important. So there are some trials that are really looking at adding stereotactic radiosurgery and radiation therapy with immunotherapy. This is one such trial that is open in my department with Dr. Rangan as the PI. He is using high doses of radiation along with nivolumab and nofenavir. This is for patients with melanoma, lung cancer, or renal cell cancer. And doing that at the same time to really looking at if we can enhance this response. And as we talk about how radiation has evolved to being much more pinpoint and meeting some of these metastases, over the last year, there have been more and more data about whether or not we should be treating patients with a small number of metastases. with radiosurgery, and instead of just putting them on systemic therapy, adding radiation. This is still an open question. This was a trial that was randomized for patients of all cancer types who had 5 or fewer metastases. Oligometastases is what we call it. And they were treated with stereotactic body radiation therapy to these metastases, as well as being on their systemic therapy. The other group has systemic therapy alone, and they showed an improved survival when you get SBRT. Now, there's a lot to be worked out about this because many of those patients also had toxicities from the radiation, and so it wasn't without some downside. But I think you'll see this area being explored more and more about whether or not we should add radiation for metastatic patients. So again, kind of early thoughts, but something that I think the field will be exploring more and more. So in conclusion, I wanted to talk about radiation and how it attacks cancer cells at the DNA level, and kind of give you a sense of how our technology is really improving to deliver radiation to a greater precision. And that makes it more relevant in places such as kidney cancer, where we're finding that high doses is really the way that we can get a response. And finally, our investigations are really ongoing about our combination with immunotherapy and the importance of oligometastatic disease. And this is where that multidisciplinary care is really important, because we work very much hand in hand with the medical oncologists and the surgeons to get the best treatment plan. So I want to thank you for your time, and I'm happy to answer any questions. Yes, I saw a hand over there.
Could you expand a little bit on proton radiation? What are its advantages and what are the applications typically?
Yes, so proton therapy, what I presented is the X-ray therapy, which is 99% of the radiation. Proton radiation is actually a different type of radiation where you use a heavy particle. So it's not an X-ray. And so the nice thing about it, it's like radiation with brakes. So basically when you shoot the heavy particle into tissue, it stops where you want it to stop. So you can stop it where you want it to stop. It's only available in very few centers, but I've been using proton therapy because I used to be in Boston at Mass General for the past 15 years. It's been used since the 1960s, but for a long time it was only available in 2 centers. It's now available in 30 centers, and we have a center in North Seattle that's the only center for the Pacific Northwest. So the advantages for it are in a lot of things. Brain tumors, as I treat, it's advantageous because you don't keep going, you don't keep getting that dose. And it's also advantage sometimes in the lung where you can kind of stop the dose. But at the same time, the technology that I presented has not completely caught up in terms of protons. And so for kidney cancer and metastases, we tend to use the stereotactic body radiation therapy because you can actually be more precise in terms of the high dose. That will probably advance, so one day protons may be helpful in these cases. Yeah, any other questions? Okay, well, thank you for inviting me, and I guess next is going to be a break for lunch. Thank you.